किया गया है।
अच्छा, तो 3-4 जो कर पाएंगे उतना कर लीजिए।
बोलो, आप बोलो, उसको बना देता हूँ। जो इसमें अभिलाष सिंह है, पूजा दोषी है, प्रदीप रो है और शिवेंदु भाई है और विकास पाटीदार है अभी।
दलीप
राखी दो।
विनेश जी मुझे मत बनाइएगा, मेरा वाइस चांसलर के साथ आज 2-3 बैठक है।
दिशा बेन आया छे कोई?
हाँ, दिशा को कर लीजिए।
એટલે આમાં ખબર તો છે ને રૂપેશભાઈ, એડમિટ કરવા પડશે તમારે બધાને, એટલે એડમિટ કરે બધાને અને એક જણ ઓલું કરે.
हाँ हाँ हाँ,
તમારે લાઈવ થઈ ગઈ ઇવેન્ટ, હું લિંક ગ્રુપમાં મૂકું છું, તમે બધાને મોકલી દો.
આમાં ઇન્ટરનેટ આજી કહું છું નથી, આમાં ઇન્ટરનેટ
બોલો કોને કો હોસ્ટ બનાવું? રૂપેશભાઈ બહાર નીકળી ગયા હતા? લિંક મોકલી છે ઓલા ચેટ બોક્સમાં, એ લઈ લો ને. મારે વોટ્સએપ નથી કનેક્ટ કરવું.
को रखेंगे, ठीक है?
ચલો, વેલકમ પ્રશાંતભાઈ. બરાબર, હવે હોસ્ટ કોને બનાવું?
રૂપેશ को.
જે કોર્સ એને હોસ્ટ બનાવું છું અને હું નીકળી જાઉં છું, હે ને?
और सर एक
टेस्टिंग कई क्वेश्चन हो तो पूछी लो.
नंजानी भी होंगी, उनके ऊपर कोर्स बना लीजिए. संध्या नंजानी मैम को भी कोर्स बनाना है. संध्या नंजानी,
वो एसएन लिखा है वो.
हाँ, एसएन.
पोस्ट, ओके. फिर
ईशा बोरा, विकास पाटीदार, दोनों को भी कोर्स बनाना है.
दिशा वोरा અને બીજું કયું નામ કીધું તમે?
विकास पाटीदार.
विकास पाटीदार.
रूम बी में करना है.
नमस्ते प्रकाश
રૂપેશભાઈ, તમને હવે
भाई.
હું હોસ્ટ બનાવીને નીકળી જાઉં છું, અને અને બીજી લિંકમાં કોણ જોડાવાનું છે? બીજી લિંકમાં
બીજી દિશા ની છે.
ये जो लिंक है ना,
બંને
इसके
જગ્યાએ
अलावा जो
પેરેલલ
दूसरी है
કનેક્ટ કેવી
उसमें
રીતે થશે એને? એટલે એ હું અત્યારે
हाँ, तो
જોઈન
दूसरी जो
કરું
लिंक
છું
है,
એટલે
हाँ
ये लिंक में से वो दिशा और विकास भाई को निकाल के बीजी लिंक में डालना है।
એટલે એ કેટલા વાગે તમારે ચાલુ થવાનું છે?
જિગ્નેશભાઈ અત્યારના હમણાં છે ને
હા.
આમનાથી ચાલુ થશે, પ્રશાંતભાઈ થી જ કરવાનું છે ચાલુ. ત્યાં સુધી મેડમ આવે છે, એકચ્યુઅલી મેડમ ને કઈક 15 મિનિટ જેવું લેટ થાય છે એટલા માટે. ઠીક છે,
એ તો ઠીક છે, પણ તમારે લિંક કેટલા વાગે ચાલુ કરવાની છે? બીજી લિંક
બીજી લિંક આમ 10:30 નો ટાઈમ છે, પણ તમે અડધી કલાક વહેલી જ કરાવી દેજો 10 વાગે.
એટલે અત્યારે જરૂર નથી ને? તો હું
ના
મેસેજ
ના
કરું. તમે
અત્યારના જરૂર
host
નથી.
બનાવી દઉં છું રૂપેશભાઈને.
હા હા
રૂપેશભાઈને
હા
હોસ્ટ બનાવી દઉં છું. ચલો, અને હું નીકળું છું.
હા, એટલે
ચલો, બેસ્ટ ઓફ લક બધા.
આમાં હોસ્ટમાં રૂપેશ અને સંધ્યા ને હોસ્ટ અને કો હોસ્ટ રાખજો.
એ તો સંધ્યાબેન તો છે જ કોસ્ટ,
હા
બરાબર.
સારું.
એટલે તમારે
અને
એમ
બીજી
જોડાશે લિંકમાં, એમાં મને કહેજો, એને હું બનાવી દઈશ. હો ને?
હા, બીજી લિંકમાં હા હું કહી દઉં છું, હાલો તમે ઠીક છે ફોન કરીને. ઓકે
હા,
હા
તમે
હા
ફોન કરજો જ્યારે જોઈન કરવાનો હોય ત્યારે, બરાબર.
હા હા પાકું
બેસ્ટ ઓફ લક બધાને.
થેન્ક યુ
भाई।
અને હા, અને તમે યુટ્યુબ લિંક પણ શેર કરી દેજો બધે.
हाँ
અને અને હું નીકળીશ ને એટલે લગભગ રેકોર્ડિંગ બંધ થશે થોડાક ટાઈમ પૂરતું, એટલે એ ચાલુ કરી દે હોસ્ટ. અને રૂપેશભાઈ,
हाँ सर।
રૂપેશભાઈ, હું જો નીકળું ને તો લગભગ બંધ થાય રેકોર્ડિંગ, તો તમે કરજો એને રેકોર્ડિંગ
हाँ
ચાલુ
हाँ हाँ,
લોકલ. હું નીકળું છું, ચલો, બેસ્ટ ઓફ લક.
ओके चलिए। रिकॉर्डिंग स्टार्ट की।
આપણે કેટલા વાગે ચાલુ કરવાનું છે?
सर 5 मिनट्स मैं करती हूँ.
જી. નમસ્તે દવે સર.
Good morning to all dignitaries, guests, delegates, and participants. With great joy and immense exultation, I, Dr. Sandhya Nanjani, welcome you all in the inaugural ceremony of Gujarat Vigyan Sammelan GVS 2022. Azadi Ka Amrit Mahotsav is an initiative of Government of India to celebrate and commemorate 75 years of progressive India and the glorious history of its people, culture, and achievements. On the footprints of central government of celebrating Indian sciences and Azadi Ka Amrit Mahotsav, Gujarat Biotechnology Research Center, GVRC Gandhinagar, in a state university, New Delhi, is organizing a national event entitled Gujarat Vigyan Sammelan GVS from 30th Jan to 5th Feb 2022. Under the umbrella of GVS 2022, a thematic conference of biological sciences and technology. I request Dr. Prashant Kunjaria, Joint Secretary and Treasurer, Vigyan Gurjari, Gujarat Plant, to brief about the event. Please, sir.
I'm audible?
Yes, sir.
Hello? Yeah. So, first of all, I would like to congratulate all the participants and host of the Biological Science and Biotechnology at GBRC, all the student member and researchers. So, first of all, let us give me little bit of brief about the Vigyan Gurjari. Vigyan Gurjari is organization. National, it is known as a Vigyan Bharati. And first of all, let us do the pratna, because I think I was not able to listen, or audio was not audible. So first of all, let us have a pratna. So Om Purnamada Purnamidam Purnat Purnamudachyate Purnasya Purnamadaya Purnamevavasishyate Ishavashyamidam sarvam Yatkincha jagatyam jagat Tena tyaktena bhunjitha Ma gruddha kasya sviddanam Vidyam cha avidyam cha Yastadvedo bhayam saha Avidyaya mrutyurtirtva Vidyaya mrutamasnute Sarve bhavantu sukhinah Sarve santu niramaya Sarve bhadrani pashyantu Ma kaschid dukh bhag bhavet Om Shanti Shanti Shanti.
Thank you, sir. I request now our Joint Director, Gujarat Biotechnology Research Center, Dr. Madhvi Joshi, to welcome our guests and give a brief overview about today's sessions.
Very good morning, all of you. It is really our privilege and honor to have today with us all the esteemed speakers and guests for the Gujarat Vigyan Sammelan, which is the first event organized in Gujarat by Vigyan Gurjari, which is a Gujarat chapter of Vigyan Bharati, and it is supported by Department of Science and Technology, Government of Gujarat. Dear friends, as you all know that Vigyan, and that too Swadeshi Vigyan, how it is important for all of us. And through this Gujarat Vigyan Sammelan, we are today trying to address some of the local challenges, trying to understand how the scientists of Gujarat and India can give some inputs for developing better scientific community and better society by using Gujarat based problems and try to solve it using biotechnology. As you all know, Department of Science and Technology is very committed for development of biotechnology, and the many aspects of biotechnology development in Gujarat are visible through establishment of GSBTM, that is Gujarat State Biotechnology Mission, way back in 2004. And today it has spread its horizon to 4 different organizations. 4 different organizations, that is Gujarat Biotechnology Research Center, Gujarat Biotechnology University, Science STBI, and GSBTM. So today, through the whole day, we have arranged various parallel sessions where you would be given exposure and you would have an opportunity to listen to the best research scholars and researchers and academicians from Gujarat and India, who would be talking on the various themes related to healthcare biotechnology, environment biotechnology, agriculture biotechnology, and animal biotechnology. So without wasting much time, before we begin, let us have a commitment that as a research community, you would be committed to use various tools that we have learned so far for solving various problems in our society, and we will bring the local science to the forefront. With this, thank you very much for joining today. I will hand over the session back to the anchor. Over to you.
Thank you, ma'am. I now request again Dr. Prashant Kunjaria sir to give few more briefing about the event. Please, sir.
Thank you. First of all, I would like to tell about Vigyan Gurjari. It has started in the 2009 in Gujarat and started in Anand Agriculture University. Now it has been spread throughout the Gujarat, and many academicians, researcher, school student, school teacher, and the people, those who are interested in science, those who do not have any scientific background in terms of education, but interested in science, are a part of this society, or you can say organization. It is a kind of voluntary organization. We have almost more than 800 life member. 20 universities are of our member, and more than 500 student members are also part of our organization, Vigyan Gurjari. Vigyan Gurjari is a part of, as Madhavi Madam told, that it is a part of a Vigyan Bharati, or Vigyan Bharati we say, Viva. It is a national organization, and more than around 20,000 scientists, technocrats, and students are member of Vigyan Bharati. We have a unit in almost all states. They are called state unit. And Vigyan Bharati is working for the two things. One thing is a Swadeshi, means our own science. So it has started in the Kerala and by Professor K.I. Vasu. He was a professor of metallurgy in Indian Institute of Science, IISc Bangalore. And he started this activity in around 82. After 10 years, there was a similar like-minded people had met at Nagpur, and they started this particular organization called Vigyan Bharati on 21st October of 1992. Since then, we have started our journey. Vigyan Bharati has a various different IAMs called World Ayurveda Foundation, which is working for promotion of Ayurveda at world level and organizing World Ayurveda Congress every 2 years. Last was held in Ahmedabad, Karnataka, in December 2018. Then it was planned in UAE, Gulf countries. But unfortunately, we all know that due to COVID, we could not organize. In fact, from the yesterday, like there was another program was organized in that 7 countries regarding the Ayurveda. But due to COVID, it was cancelled. It was also organized by Ayush Mantralaya was involved in that, and Vigyan Bharati was also there, part of it. So we are working for that, like Ayurveda. We are having one another global arm, which is called GIST, Global Indian Scientist and Technocrat, where we try to get give the platform to the people, those who want to contribute for India, and those who are settled in other countries or citizen of other countries but have some feeling for the India. So likewise, that work has also started in US and in UK and Canada, and slowly it will spread throughout the world. So we wanted to have a best utilization of these people for our countries, and if they want to contribute more, they can also, we would provide at least some facilitate or platform to them for this particular work. For a school student, because we have increment, we are seeing that school students are actually afraid of science, and we know that science, to perform in science, we need some scientific education properly, and then only. So in schools, many people opt for some other subjects due to fear regarding the science, and feel that India is nowhere in the science. So to make them proud, we are organizing every year Vidyarthi Vigyan Manthan event, which is a school their online platform. We invite students to give the exam. Almost 1 lakh students or more than 1 lakh students are participating each year. Since last 6-7 years, we are organizing that this exam, and there we have designed our syllabus a little bit in different ways. There we have incorporated contributions of our scientists, pioneer scientists of India, and every year life stories of 2 scientists, and one book Vigyan Bharati has prepared, that is a part of the syllabus and their normal curriculum. So likewise, they come to know about the scientists. Those many times it happens that we know only one paragraph or few lines about one scientist, but what exactly really they have done, and in which condition they have done what was the motive, and all these things matter. So we expose them to this particular all events which has occurred in the past, and so how India has started is performing in modern sciences. At the same time, we also expose them to our ancestors like Rishi Munis, those were also scientists, and gave the lot of principles and how they have worked in, what are the books they have written. All these informations we try to give them in this particular exam. It is conducted in at 3 level. first is a school level, and then based on merit we give rank to the in the district level also. Then from all different standards like class 6 to class 11, we call almost 20-21 student from the throughout the state and organize one state camp where they have to come. So from there we give the cash prize also to all one class rank or 1, 2, 3. And then from there, first and second rank will be selected for the national camp that we generally organize in some national laboratory or some places where they get again some encouragement for the science and all. So this is regularly we are organizing. And but the last 2 year we were unfortunate because of this COVID. We organized all event on online platform. Otherwise, level and state level and national chemical. But now it is on for virtual mode we are doing. Likewise, we are having emails. We are having Shakti Ayam. Many like women scientists. So we are also promoting women scientists also. And India has produced a number of women scientists, and they have done pioneer work in their field. So we also include their stories in VVM also. And separately, we have one particular arm where they are working. For Ayurveda, as I told, that Ayurveda foundation is there. We have Nasya also, where Ayurveda students are part of that, and they also conduct various. They are also, work of awareness of about the Ayurveda in our society. We due to the efforts of Vigyan Bharati in 2014, we have seen that after our SSV Pradhan Mantri Sri Modi ji separated health department from the Ayush department from the health, and made a separate ministry called Ayush, where we are promoting Ayurveda, Yoga, Unani, Siddha, and likewise. So all these 5 different therapies which are there in the societies, and more and more scientific studies are conducting. So we have seen this is a part of the World Ayurveda Foundation and Nasya. Similarly, we are having another organization called Vibha Vani. Vibha Vani is a Vibha associated NGOs. Generally, we are called them NBOs, National Building Organization. So we have seen that in all the sectors like health, like education, like awareness about the government policies, related to many organizations are related working on for the improvement of the land and pollution and a lot of things. So we are providing platform to them, and Vibha Vani is working for them. So we are like connecting all these NGOs, those who are working for our countries at various level at various places. So that is what we are doing. So likewise, there are many things which are happening. Every state has their own unique programs where they are giving a very good impact on the students and community of science as well as the common people, because more and more people if join this like movement, they our intentions whatever are there, they will get good results. So that is why we are like call people. We are also organizing Bharatiya Vigyan Sammelan to promote the Indian language, because many times it has been seen that in country most of the students understand and get educated in their own languages, but when higher education comes, they have to go for English as a medium of understanding. So there we have seen that and it is study also that if student understand or learn in their own language, or we can say matrubhasha, then they can explain it very well. and we have seen that English is only a medium of expression. But when you want to dream about something, about some idea and working, then our own language is preferred. So, and many people, those who have studied in their language, mother tongue, and have done very good in science also. So there are all kind of examples. So to promote them, we are conducting every 2 year Bharatiya Vigyan Sammelan BVS. We are also conducting some programs called Vishwa Ved Vigyan Sammelan. It is throughout world, many people are working on the science, Vedic science and related work. So to provide them platform, Vishwa Ved Vigyan Sammelan is organized every 4 year, where we invite people throughout the world, those who are working on based on Veda. Veda means Vidya, nothing. So people see it as a some religion book or something or some other thing, but actually it is a knowledge. And those based on that knowledge, if people are working, we are inviting all kind of people, those who are working on it. Likewise, we since last 6-7 years, we are conducting IISF. It is called India International Science Festival. IISF is a very big event organized most of the time with cabinet, like Central Ministry of Science and Technology, Earth Science and Health and all. And all the time, different events are organized in that. We are also again promoting a lot of things through IISF also. And people every year, it is organized in some cities where students of all schools and common men visit there. We are having expos also there, like DRDO and other ministries also take part in that, and all the labs of Government of India, like CSIR lab, National Institutes, and various organizations display their work there. And ultimately, so this is what is been done. So this is mainly about the Vigyan Bharati, and you can join with us in our work. And this is what about the Vigyan Bharati. You can contact us. We have website also, vigyangurjari.org.in. And thank you for giving me the opportunity to explain about Vigyan Bharati. I thank all organization and congratulate and best wishes for the event. Thank you.
Thank you, sir. We should now begin with our first session of the day, that is panel discussion on contribution of biotechnology in Atmanirbhar Bharat. Now I request Mr. Utsav Parbar, IAS officer and deputy director of Doordarshan Ahmedabad, to please moderate our first session. Please, sir.
Am I audible? Hello. Yes. First of all, I would like to welcome all of you at Gujarat Vigyan Sammelan, which is being organized on the theme of biological sciences and technology. And today we are beginning with the first session of the day. It's a panel discussion on the contribution of biotechnology in Atmanirbhar Bharat. So I would like to thank Dr. Madhvi Joshi, Dr. Chaitanya Joshi for inviting me as a moderator. And I hope that all the speakers will put their remarks in time, because we are running tight in the schedules. But first of all, I would like to invite our eminent panelists, Dr. Arun K. Rawat, Dr. Sunil K. Raghav. Then we have Dr. Snehal Bhaktharia and Mr. Akash Bhausar. So welcome all of you. I would like to give a brief intro of all the eminent panelists that we are having on the panel. Dr. Arun Rawat is a senior scientist and advisor in Department of Biotechnology, Ministry of Science and Technology, Government of India. He's also involved in various scientific advisory committees and policy making committees. Today, we are going to discuss about what are the policy prescriptions for the biotech sector. Welcome, Dr. Arun K. Rawat.
Thank you. Thank you very much. Can you hear me, sir?
Yes. Yes. Yes.
Okay. Thank you very
Then
much.
I would like to welcome Dr. Sunil K. Raghav. Sunil K. Raghav is a scientist staff in Institute of Life Science, Bhubaneswar. He has received master's degree from CCS University, Meerut, UP, and PhD from IGIB, Delhi. He has completed his postgraduate from Center of Biotechnology in Finland, and has also worked as a scientist in Swiss Federal Institute of Technology in Switzerland. He has very much various exposure and experience in fields of genomics, especially integrative genomics, to decipher the molecular mechanisms which is involved in immune response. So I would like to have Sunil Raghav, Dr. Sunil Raghav on the panel, and we'll discuss about the probability and the potential of genomic sectors in India for Atmanirbhar Bharat. Welcome, Dr. Sunil.
Thank you. Thank you very much. Yeah.
Yes, yes. And then we have our own Dr. Sunil Bhaktharia, who is Joint Director, Gujarat State Biotechnology Mission. He has completed his academics in Saurashtra University and is presently working as Joint Director in GVS-2022, a nodal agency for implementing all biotechnology-related policy matters and innovation-related things in Gujarat. And he has played a key role in establishing several research initiatives in the state. So he's running various network mode program on cancer biology, antimicrobial resistance, and water management and genomic research through GVS-2022. I warmly welcome Dr. Snehal Bhaktharia in the panel.
Thank you, Utsav.
Yes. And then we have very young and dynamic Mr. Akash Bhavsar, who is MD of Sky-Quest Technology Group. He has done his postgraduate diploma in production management from Indian Institute of Planning and Management, Delhi. He's a group MD in the Sky-Quest Technology. And it's a company which deals with the global technology aggregator and accelerator, working in the areas of agri-tech, healthcare, life science, nanotech, clean tech, and ICT, founded in 2006. So they provide technology-based solution to the ideas that innovators and the young minds think of, and they make it realize in the real life. So Mr. Akash Bhavsar, very warm welcome to the discussion.
Thank you, Utsav, for the introduction. Thank you, everyone.
Yes. So I would like to begin with Dr. A.K. Rawat. Dr. A.K. Rawat, I would like to ask that India is among the top 12 destinations for biotechnology in the world, with approximately 3% share, if I talk about the global biotech industry. India is also a very key contributor in biotech sector. When we are aiming for $150 bioeconomy by 2025, and when India's overall holistic vision is to reach to become a $5 trillion economy by 2024, what is your policy prescription for building a vibrant biotech sector, Dr. Rawat?
So thank you very much. Let me go through the background of the biotechnology in India, development of biotechnology in India. See, it was started long back in 1983. At that time, biotechnology, actually, the first time in India, biotechnology department, it was not department actually, it was a part of the Ministry of Science and Technology, and it was established in a small section of Department of Science and Technology in the form of National Biotechnology Development Board, NBDB, and it was the foresightedness of our science policy maker at that time, because only 2-3 countries in the world, they started small departments in their science and technology departments. So India also started in the form of National Biotechnology Development Board. And in 1986, it was separated from Department of Science and Technology, and full-fledged department under Ministry of Science and Technology was developed. And you can see during a very short period, this department has done tremendously well in all spheres of life. Basically, biotechnology, it overlaps the activities of almost all science departments, including Department of Science and Technologies, CSIR, ICAR. So all departments, we are working actually in the area, in the environment where we are overlapping various activities, including ICMR. So biotechnology during this period has shown its presence in all fields of life. So actually that now that biotechnology, all biotechnology products, all biotechnology research is being monitored by the Department of Biotechnology. And there is a 3-tier system in that. This is a very well developed system. The first of all biotechnology related research will be considered in IBSC. IBSC is Institutional Biosafety Committee. And from IBSC, once a research project, a research problem is cleared by Institutional Biosafety Committee, it will go to the RCGM. RCGM is a review committee on genetic manipulation. This RCGM, DBTs hold this RCGM, which is independently chaired by an eminent scientist. And if a project is cleared by RCGM, then it will go to GAC, that is genetic that comes under Ministry of Environment. And this is the agency which gives the clearance of the production of that particular product or particular crops. Genetic engineering, this is known as Genetic Engineering Appraisal Committee. And once it is cleared by, recommended by this, the government gives the final clearance. I will give you an example of BT cotton. In India, we have only approved one crop, that is BT cotton. And this cotton, this particular product, particular crop, was basically approved in 2000, sometime in 2004. And just see, in 2019-20, almost 98% of crop is BT cotton. And the production has actually, the total crop production has increased almost 6-7 folds. That number of cultivative area has increased tremendously. So this is only this, why this crop has been adopted by the farmers. What is the reason? Because it is giving high return. That basically you can say that very less amount of pesticide is being used, and fertilizer is also being used in less amount, and the crop production has increased tremendously. So these are the benefits of the biotechnology in India. So I gave you one example because you can see that how the biotechnology products are being adopted by the end users. Similarly, you can see here that in case of vaccines, medical science, so India is largest producer of the vaccine in the world. And our government has given a lot of emphasis in the health sector. This is very, very important because that the emphasis, the promotion given by the government in health sector and using biotechnological tools has actually attracted a large investment in India. You can see this even in COVID duration, the Department of Biotechnology cleared the guidelines for COVID research within 3 days, and our scientists, our industries started working immediately, and we could produce large number of vaccines in India. So these are so biotechnology is actually is giving a lot of emphasis, a lot of
Yes, we can take a short break, Dr. Rawat, and then I'll
please.
come back to you. I'll go to Dr. Snehal Bhattaria. Dr. Snehal, as you are overlooking the holistic view of this biotechnology sector in Gujarat, I would like to know that what are the major sectors of biotechnology are being explored by Gujarat biotechnology sector, and what is the scenario? Where does Gujarat stand in the national scenario of biotechnology? Dr. Bhattaria.
Yeah, thank you. Thank you, Dr. Utsav. So biotechnology in Gujarat started in early 40s. If we see in terms of industrial development with companies like Alembic, there was a slow growth from 40s to 80s, where we see near about 18 companies in Gujarat. At present, in biotechnology, there are total 232 companies at present. And if we look in terms of number, we have agri-biotechnology is leading with near about 83 companies, followed by pharma biotechnology, which is having near about 75 companies. There are some bioservices and bioindustrial companies in this segment, key companies. One of the panelists is with us, SkyQuest is into services. If we look into the revenue segment, number is one part where agri is leading. But if we look into revenue segment, revenue is majority of the revenue is coming, more than 40% of the revenue is coming from pharma biotechnology. 20% revenues are coming from agri biotechnology, followed by bioservices, environmental biotechnology, and other smaller companies. If we look at some of the key products in pharma biotechnology segment, so biotherapeutics is a leading sector for Gujarat, where we lead in India also in terms of highest number of biotherapeutics approval in the country, whether it is biotherapeutics, biosimilars. Our companies have two models. One is in-house development, where smaller or mid-level companies are focusing on in-house development and R&D. Bigger companies, namely few like Sun Pharma, Zydus Cadila, Intas, Torrent, they acquire smaller companies from Europe. They also work in advanced technologies such as gene therapy. If we take example of Intas, they have a dedicated team in this advanced area of gene and cell therapy, a dedicated team of 45 scientists working. And DBT has recently declared the gene therapy guideline in 2019, and slowly we are also coming up in Gujarat with some of the gene therapy segment. If we look into the vaccine and COVID response, our companies like Hester Biosciences, which was a leading company in poultry vaccines, diversified in collaboration with Bharat Biotech and GBRC for manufacturing of the COVID-19 vaccine. If we look at a smaller segment, considering the Atmanirbharta, earlier we were importing, there has been a lot of import still in RT-PCR and other diagnostic kits. But, I'll give, there are few wonderful examples where, PG and PhD students, in some of the universities came together, and now they are also supplying the ICMR approved COVID-19 kits. So there has been a lot of, inputs. State government is also, since the time of, then the Chief Minister and now Honorable Prime Minister Modi Sahab had given, a slogan when the biotechnology mission was established, that BT plus BT is equal to BT. Bharat today plus biotechnology is equal to Bharat tomorrow. And then state government started working in three core areas. One was business development, where there has been a dedicated support for establishing biotech park, establishing biotech companies, establishing biotech incubators, contract research support scheme, and establishing finishing school to generate industry-ready manpower. Slowly, biotech mission was segmented into various dedicated organizations like, Gujarat Biotech Research Center focusing on research activities, Savli Technology and Business Incubator jointly established by Department of Biotechnology, Government of India and Government of Gujarat, Science and Technology for incubation support, Gujarat Biotech University. So this is how state government is also supporting the ecosystem, where dedicated support is for industries. Why in Gujarat, just to share the details, biotech sector was the sector which is having highest realization in terms of investment as compared to any other sector in Gujarat in biotechnology. So this is in brief, state government is trying to do, and this was one of the first organization established as a nodal agency for biotechnology in the country
So,
by state government.
yes. Wonderful and apt response that you've given, Dr. Bakhtaria. That reminds me the clarion call of Jai Jawan, Jai Kisan, Jai Vigyan, and Jai Anusandhan. And with this clarion call that has reminded us in the past and the glorious future that we are going into, I would like to ask Mr. Akash Bhausa, Akash, you work with United Nations Sustainable Development Goals, and they're so crucial for the overall development of the humanity. You often say that innovation and entrepreneurship can accelerate the rate of achieving SDGs. So how is this true for the entire biotech sector?
Thank you, Utsavji, and I would like to thank the GVRC team for organizing this, and Madhviji and Snehalji for inviting me to contribute my inputs and insights. So, Utsavji, to your question, you see, sustainable development goals, if one gets to look into it, it has, there are 17 sustainable development goals. And if you look at all the primary goals related to food, agriculture, food and agriculture, healthcare, even water, water affects almost 6 to 7 goals. SDG 6 is related to water, but it affects almost all goals. The partnership for goals, the goal number 17. Primarily, sustainable development goal enablement, biotech has a massive role to play. As we know, there are various fields within the biotech or the application areas of biotech. It's far-reaching, I think. So that enables, so any progress in the biotech sector is actually a force multiplier towards a nation or us all as humanities towards humanity's progress towards achieving these SDGs by, there are lofty goals, of course, to achieve them by 2030. In terms of in the wake of pandemic, the biggest testimony to the role of biotechnology is the quick development of the testing kits and the vaccines and for the advanced, you see the molecular antibodies which have played an important role, like tocilizumab, bevacizumab, and some of these have been also their biosimilar versions being developed indigenously in India. So it has played an important role in keeping the spine of our healthcare system, in terms of providing this so much critical support needed at times and at speed, which we probably, all of us from the industry, couldn't even think of that we were capable of doing so. But I think the good understanding and skills and the infrastructure we have has all come together. And there is tremendous potential of how biotech can play a role, for example, in food security. We are seeing a lot of increasing use of biotech in from a food security standpoint, where animal protein becomes a very important segment. So we are seeing more and more plant-based alternatives, biotech-driven, like we have the cell-based, lab-based meat, which actually, as we know that, let's say 1 kilogram of any average meat would require 10,000 liters of water. So it is extremely resource intensive. The consumption of consumption of meat, and which globally has a massive implication on the greenhouse emissions, on what should be served as food versus feed, And so all of these debates can actually be solved to a great extent by use of inventive and innovative applications of biotech. And we are seeing several such real examples. I'm just recently involved in a very interesting company called BF Like from Netherlands, which we are trying to bring to India, which is basically fungus-based alternatives to meat, and they've been able to recreate animal blood and everything in its complete. I'm a vegetarian, so I can't say much beyond this, but on the science side of it, it's extremely potent. And so yeah, I mean, there are tremendous examples of how biotech will actually just biotech along with IT. I mean, they are the force multipliers, right? So, and technology, and so I think the session is very apt to discuss and in fact applaud the what we have achieved and what we should work towards.
Yes. So because the time demands the solution of the questions that entire humanity is seeking, and I think we all are here enabler to solve the questions which will define the human future. Now we have Dr. Sunil K. Raghav with us. Dr. Raghav, when Watson and Crick found the building blocks of life, DNA, it revolutionized the biological sciences. Understanding of genetics expanded our horizons to understand human and the natural world. And during a pandemic, we have already seen the major role played by the genomics. My question is to you, sir, that how this knowledge of building blocks of life can lead us to the building blocks of self-reliant nation, that is Atmanirbhar Bharat.
Yeah, thanks, Usav. So thanks GBRC and all panel members, because it was like, I would say it's very important discussion, because now we are at that stage in last 5 years or 6 years, we are building toward more like as a community biotechnology, which is taking a boom of growth. And these discussions are very important, and to motivate also the young mind to really bring them into the mainstream. And as Prasant sir was saying from Vigyan Bharati, that IISF, I would say they're playing very important role in bringing common people, students, and making them motivated to bring them into this, to really understand what is going on. Because when they will be motivated, there will be a lot of things coming up in biotech. Okay, so in terms of genomics, as Utsav said, I would say that COVID pandemic, definitely it was a problem, but it helped us to really understand what is the importance of genomics or molecular methods to really understand the diseases or diagnostics, and building that infrastructure and putting that in our country, because we are reliant on a lot of kits and everything from outside. And that clearly showed us during the start of the pandemic, as you realize that Thermo was providing kits, because outside the pandemic was much more, so they were not able to provide the kits even in the start. And we were really like in problem that we cannot do the diagnostic testing for our person because we don't have the kits. And it took some time in the start to really build our kits and then go ahead with it. And it created problem also in the start that we didn't have the kits. So the thing is, I would like to say that genomics is definitely coming up as a major sector in diagnostics in medicine, and even in biotechnology sector, as basically nowadays we are looking into the plant genomics as well, to identify the drought resistant genes and all, to make recombinant, as Dr. Rawat said, like BT cotton. Because when they will be interaction with the common people, they will understand that this is required in future. So then they will understand that the recombinant things, recombinant DNA technology will definitely help us to come up with the new findings and new methods to give better crops or better diagnostic methods. So it's now the fields are combining. It's not like before genetics was separate, all bioinformatics was separate. Now we are bringing everything together and finding the solutions. Now I will just give you an example of like we just started 2 years back the Genome India project, you will be surprised to see that we don't have a reference map of India for genome for all the population. And we are still relying on Caucasian population, European population to do all the diagnostic assays. And that is surprising, no? Because our population is very different from Europe. But when we detect cancer or other immune diseases, we use their reference. We don't have any reference. And our population is very different. South population, East population, West is very different. We cannot use that reference map. So we started 2 years back the Genome India project, where there are 22 different institutes coming together from all over India. And then we are sequencing whole genomes of different populations. And the idea is, if we can map reference genome of our population so that we can develop diagnostic kits, arrays, population specific arrays. And this will definitely boom a lot of industries coming out. The reason is because then population based assays can be done different, as you can see these PCR based assays for COVID. Similarly, population level assays can be developed using that map. And that is just a simple example. Similarly, for cancer therapeutics, currently we don't use genomics. It's more of a trial-based methods. Doctors, they just give the medicine, they see if it doesn't work for 3 months, they change the medicine. But there is a possible way to do pharmacogenomics at a personal level and really identify which drug is going to work. And there are companies coming up now, but the number is still less. If you see our population, the number of those companies are just few. And that is not going to help. So the current scenario of really building these startups, incubation centers and all, several companies are coming, and definitely this field is going to boom like anything in both diagnostics, medicine, as well as for this crop related, plant related things. So these things are coming up. So that's the thing in genomics that I would like to say.
Yes, Dr. Sunil Raghav, as you said, that we are ushering the era of quantum biology, where we are integrating, like micro is new macro. And when you are talking about recombinant technologies, like recombination of our ancient knowledge, ancient traditions, ancient knowledge with future and present understanding of the world will definitely lead us to a new era. Now I'll come back to Dr. Raghav. Dr. Raghav, I would like to say, as Dr. Sunil Raghav, sorry, Dr. Rawat, I'm sorry, Dr. Rawat, as Dr. Sunil Raghav has said that we are proving our skills and specialization in genomics in India, and we are also strong in manufacturing and generic drugs and leveraging that prowess to produce specialty drugs, vaccine, and biologics. To achieve this global leadership in biologics, we need to enhance the capacity, competency, and infrastructure available in the country. I would like to ask Dr. Rawat that how do we fine-tune our policy with the investment risks associated with the prolonged research period and the commercialization of the product? Dr. Rawat.
So as far as this promotion of research in biotechnology sector is concerned, see today, when I did my MSc and PhD, at that time, no engineering colleges, no medical schools were working on biotechnology aspects. Today, you go to any institution, whether it is AIIMS, whether it is IITs, they have biotechnology department, separate biotechnology or biological science department, where they are working on biological sciences on this aspect. So government has given a lot of emphasis on human resource development and infrastructure development. So one thing is very, very clear, that government has done a lot of work and a lot of emphasis is being given. Even in traditional institutional, you can see in ICR also, in ICMR also. So we are all scientists, most of the scientists are working on various aspects of biotechnology. So that is one thing. As far as government government support is concerned, so today, as you told earlier, India is counted among the top biotechnological destination in the world. And we are third largest biotechnological destination in Asia Pacific region. The country today have second highest number of US FDA approved plants after the USA. So USA is the first and India is second, which includes 66, almost 700 US FDA approved plants outside the US, and total 44% of global abbreviated new drug applications and over 1,400 manufacturing plants with the World Health Organization. So this is the strength of India, and we are continuously actually building on these basic foundations. The Indian biotech industry as present have approximately 3,000 startups. This is a big number. And you see that last 2 years, 20 and 21, both, that this was the challenging year for the whole world, not only for India. And India has come up very nicely, as Dr. Raghav was also told, very nicely India has come up. Initially, there were some problems, but within few days only, even not months, we came up with the solution in diagnostic kits, even vaccines, different type of vaccines. So this is the strength of India, and it is expected that by 2025, India will reach approximately $150 billion. So this is at present it is around 80. So within 2-3 years, had it not been COVID 2021, we might have already gone several steps further. But there is a need from the government side. There is a need actually, at least in agriculture, there is a need to see how we can bring some more biotech crops in the country. We just see that India worked basic research on this BT brinjal. A large number of brinjals, a variety of brinjals were developed in India, but since our regulations are not allowing to approve this crop, Bangladesh has taken advantage of this, and Bangladesh is now supplying brinjals to India. Even we are taking this soya oil. So we are bringing soya oil from outside country, yes, which is basically the biotech product, but we are not producing these crops here. So there is a need, actually the government now, since I am superannuated, yes, now, so I can say now, so the government should see all these things. We are ready, our scientists are ready. All these, simply we need the approval from the government.
Yes, Dr. Rawat.
If it is there, then we will be go ahead.
Yes, we're running short of time, so obviously that we could have more of you. But now I'll have just quick comments from Dr. Snehal Bhaktharia that Gujarat has officially stated that biologically to resolve and continue endeavors for socio-economic upliftment, biotechnology is the potential tool for development. How is this dream visualized, Dr. Bhaktharia?
So there are 3 areas, as I indicated earlier. Business development is one area where state government is focusing. Human resource development is another area, and research support is a third area. Through these 3 actions, we are trying to achieve this dream. So in a way, if you see in terms of policy support, this is one of the best policy in the country, where we are supporting various segments of biotechnology development, such as for Biotech Park, we have a support up to 25 crores to the tune of 10% of the capital subsidy. If you look at the unit, we have a support up to 50 crores per unit to the tune of 10%, and investment interest subsidy is support is 1 crore per year. So this is how it is supported. In terms of research support, there are several network programs initiated I would like to, state that, long back, a first bacterial genome of the country was sequenced by Gujarat under the initiative which is known as Gujarat Genomics Initiative. Key things like a pride species of Gujarat lion, where, the mitochondrial genome of Asiatic lion was sequenced by Gujarat. State has initiated several network programs, namely few is network program on cancer research, where we are focusing on, population, our own population. We have a program on environmental biotechnology and allied area network program. Considering, as Akash mentioned, SPG, antimicrobial resistance is one of the key area which is also included, and it is considered as a developmental threat globally. And it is predicted as one of the major challenge, even, major challenge than that of the present COVID challenge.
So
Where we have
realizing the vision to the land is very important. I would just like to have one question from the audience then, because time is not permitting, and all. I have just 4 wonderful guests like with ocean of knowledge, but I would like to have some questions from the audience, and then we can close the session. Mr. Chandresh Mehta is saying that hopes are high from GMO with increasing population and scarcity of resources. As Dr. Rawat mentioned in his talk. The last question that I can ask Mr. Akash, that what are the opportunities for the young people for the startups in biotech sector? And meanwhile, if someone has any questions, they can ask me. Otherwise, after that, we can close the session.
Thank you for the question, Utsarji. I think with regards to the opportunities, especially for startups, and given the wonderful policies and the support schemes which are available with the central government as well as Gujarat government in specific, I think diagnostics is in a very important area where we would basically there are tremendous opportunities for startup molecular biology and molecular. I mean, the diagnostics based on genomics as well as. We also see a lot of potential in doing process development innovations related to biosimilars. Also in the food sector and the agri sector, dairy, there is tremendous applications of biotech, and those areas are ripe for, I would say, disruption and need for great solutions. So this is something which also I think there's a question on precision medicine. If it's okay, I'll take it. I think the combination of a targeted therapy as well as better imaging and diagnostic solutions, that is a very potent way of solving several currently unsolvable or challenging diseases. So that's there. In Gujarat, I think the landscape is very very nascent right now for precision medicine. But broadly in India, there are a lot of good technology companies emerging, biotech companies emerging, working in these areas.
Yes.
Thank you.
So wonderfully that we are doing. Dr. Raghav, final remarks from you, and then conclude the session.
I think this is the question that I was saying, that precision medicine definitely is going to be a really big sector, and that's coming up and that is very much required. Because outside it's already going on. Now we are high with genomics. A lot of expertise came out already at different areas because of COVID. Nowadays, even hospitals have the genomics facility because of COVID. So I think it would be really good to go ahead with the precision as soon as possible. And that will really definitely help in Indian scenario. That's very much required. Thanks.
Yes. Thank you so much, all the panelists, Dr. A.K. Rawat, Dr. Sunil Raghav, Dr. Sunil Baktharia, Mr. Akash Bhavsar. All of you have given the beautiful insights of the world of the biotechnology, as we all are connected with the vision of Atmanirbhar Bharat and the happy world, Vasudhaiva Kutumbakam. I think this discussion will enhance the knowledge of the viewers and the participants who are all over here. I would like to thank Dr. Madhvi Joshi, Dr. Chaitanya Joshi for giving the opportunity to conduct the session. And that's all for the day, because lots of things and events are that's following this event. Thank you so much.
Thank you to all the panel. Thanks.
So Now, for upcoming sessions, we have organized 2 different virtual rooms. I request all the participants to join the respective links. Delegates who want to join virtual room A should stay on the present link, and those who want to join virtual room B can join the other link provided in the schedule. The link is also provided in the chat box. In virtual room A, we have organized talks and presentations associated with human and animal healthcare as well as plant biotechnology. Whereas in virtual room B, we have presentations and talks related to industrial and environmental biotechnology. So in the meanwhile, we can take 5 minutes break, and the people who are interested to switch in virtual room B can switch to that. Welcome back all. I, Dr. Sandhya Nanjani, on behalf of GVS 2022, welcome you all in the virtual room A. For our next session, we are privileged to have with us Professor Alok Dhawan, Dr. Bakulesh Khamar, Dr. Subir Majumdar, Dr. K.G. Tirumurugan, Dr. Mangal Singh Rathore, and Dr. Rukam Singh Tomar. I would like to invite Professor Alok Dhawan for his talk on nanotoxicology, a journey from laboratory to policy. Before beginning, I would like to take this opportunity to introduce Professor Dhawan. He has completed his PhD in biochemistry from Lucknow University and currently working as director at Center of Biomedical Research, CBMR, Lucknow. He is also founder director of Institute of Life Sciences, Ahmedabad University, and previously worked as professor at Department of Biotechnology, Assam University, Silchar. He was also visiting scholar at Michigan State University, USA. So was principal scientist at Indian Institute of Toxicological Research, IITR, Lucknow, UP. And also has been Boy Scout Fellow at Directorate of Medicinal and Aromatic Plant Research, Boriavi, Wales and Bradford. Sir has 7 patents, 6 copyrights, and more than 150 publications to his credit. He has been a recipient of Young Scientist Award, Vigyan Ratna Award, Institution Development Award, and many more. I would now like to request Professor Alok Dhawan to please deliver the talk on nanotoxicology, a journey from laboratory to policy. Please, sir.
Thank you very much indeed. I'm just going to share my screen. That should be good. Can you see the slide?
Yes.
Okay, so just a minor correction in my introduction. I think one, that I was formerly the director of CSIR IITR. And secondly, I have never worked in the area of medicinal aromatic plants, which was mentioned. I think it's perhaps my colleague from CMAP who's worked, so I've not been there. But other than that, I'm very glad that I've been invited to speak on this occasion. I must thank Vigyan Bharati, GVRC, Madhvi Joshi, and the entire organizing committee of the Gujarat Vigyan Sammelan 2022 for giving me this opportunity. I think I was also attending the previous session where we were talking about contributions of biotechnology in Atmanirbhar Bharat. And I feel that this is another area where India has actually worked a lot and contributed a great deal, and that is nanotechnology. And I think it's very befitting that I share with you some things that were done when I was at IITR. I started the field in somewhere in 2005-06 in India, and then we have risen to where we are now. So I'll just share some of my experiences with this. Currently, I'm at the Center of Biomedical Research, as mentioned. This is an autonomous body under the government of Uttar Pradesh, and perhaps the only institution in the area of biomedical research being funded by any state government university, state government for research. So just for students to understand, what I'll be mostly dwelling on, mostly discussing, is the fact that when you talk of nanomaterials, they are very small structures. So it could be of the order like a buckyball, which is only 1 nanometer in diameter. But when you look at the biomolecules such as DNA or so, they are approximately 2 to 2.5 nanometer in diameter. So which means that it can, even if the nanomaterials are inert, they can actually physically interact with the macromolecules, and that is where the concern is. So this gives you a larger picture of the dimensions of various things, but I would like you to just concentrate on these 2 things. And with this in mind, we looked at various aspects of toxicology, and we essentially gave an hypothesis that as you go down in the surface area, go down in the size, the surface area increases, and so does the reactivity. So it's good for catalysis, but it's not very good for biological systems. And Andre Nel, another scientist, he came up with similar theory in 2006, where he mentioned that if you go down the size to about 5 nanometers, 60% of the molecules are at the surface, again making it very reactive. So the same substance that you're using in bulk form, when it is converted into nanomaterials, might become hazardous to the system. Now, as far as the materials are concerned, right from production to end of life, at each point in time, they get released into the environment, and ultimately they become part of the environment. So not only the person who is in the manufacturing process or the consumer, for example, is getting exposed to these nanomaterials, but also the environment is getting exposed, for which it has no meaning. Means nanomaterials are not intended for the environment, but because they end up in all the entire life cycle of this material, they end up in the landfills or incinerations, etc. So it becomes part of the environment. So therefore, environmental exposures are equally important. So when we started nanomaterial toxicology or safety, it was very pertinent for us to understand what all are the major issues arising out of the nanomaterial. First that we discussed was the coating during synthesis. So when you synthesize nanomaterials, several times you use solvents that are required. If you're doing it physically, it is fine. If not, then if you're using solvents, you need to consider the coating of the solvent as well. Of course, characterization, visualization, dosimetry is again very important, because on the one side, we generally use microgram per ml or milligram per ml kind of systems. But here you can also use the surface area as well as the particles, number of particles per surface area. Agglomeration was another issue, wherein the particles actually agglomerate and become bigger size particles. Positive negative control means we do not, in chemical toxicology, we have negative control and positive control, but in particles, we do not have negative and positive control particles even now. Right? So we have to do with chemical when it comes to putting control in the experiment. Polydispersity is another issue, which means that there are different sizes of, if the suspension is not uniform, then there are different sizes of nanoparticles which make it polydispersed. So then it is difficult for you to say whether a particular size of nanoparticle is causing the toxicity or it is the bigger nanoparticle. And same is true with exposure assessment. It's very difficult because of the size to assess how much a person is getting exposed, unlike radioactivity, for example, where you have meters and you can actually monitor the radioactivity exposure. And this is not enough. We still have other issues such as size and shape of the nanomaterial, in vitro versus in vivo extrapolation, life cycle analysis, dosimetry, and so on and so forth. Ecotoxicity, as I mentioned, because they are getting in the environment. So ecotoxicity and environmental monitoring are equally important. But also what was important was to see whether these nanoparticles interfere with the toxicity assays that we are doing. So therefore, development and validation of test models was also equally important. And ultimately, it was realized globally, and European Union actually came up with a flagship program only on this development and validation of test methods for toxicology. So unlike the chemicals, the nanoparticles are not universal. So even if we are doing zinc oxide or titanium dioxide nanoparticles, the toxicity of them will change if we change the size and shape, right? Unlike, let's say, if you're doing toxicity of ethyl alcohol, methyl alcohol, it will remain the same. So that creates another issue, especially with the industry, that once you change the size and shape or the coating of the nanoparticle, you have to do the entire toxicity all over again. Right? So that is another issue that is coming up. I'll share some examples with you that we have done. It is not, I would say, very detailed in terms of data that I'm going to show you, but give you an overall view as to what we have done and how it helped in the policy formulation. I think that was another area which was dwelled in the previous session as to how our own country should have our own policies that will help in becoming Atmanirbhar. So same is true with nanomaterials, and there was a major issue raised by the industry since we don't have any guidelines. So how do we progress with our development of product? So in this case, I'll just share with you the effect of zinc oxide nanoparticle exposure on human epidermal cell lines. Again, for the students and young faculty, I've just mentioned, please use models which are relevant to your experiment and not what is available in your laboratory, right? So here, because zinc oxide is used in sunscreens, we have used human epidermal cell line to begin with. Okay, so as I mentioned, characterization was done using 3 different methods: transmission electron microscopy, Brunner-Emmett-Teller test, which tells you the surface area, and the dynamic light scattering, which again tells you what is the impact when you make suspensions, how big is the nanoparticle, and so on and so forth. So these are the what you get out of the measurements means BT was given by the manufacturer. We did our own size through electron microscopy, and we also did the DLS, dynamic light scattering, which is the hydrodynamic diameter for zinc oxide. And then we looked at morphological studies. So these are very interesting findings. If you look at the when you look at control cells, they are fine, they're adherent cell lines. But as we increase the concentration of nanoparticles, the cells actually get damaged and they float. But when you wash them off before doing tests in the cells that are left, so one feels that, well, these cells are quite happy. But if you look at the microscope, you will actually find that a lot of cells have died. So this was something that we put in our paper just for the others to understand that while we are doing such experiments, we should also be cognizant of the fact that we need to see whether the cells are dying, and if so, how many cells are dying and what are left in the flask. Now the issue was that whenever we are doing electron microscopy, it is very time consuming, and not only that, very small number of cells you can actually click photographs and assess them. So although it gives you an idea as to where all the nanoparticles have gone, so we could find out that, for example, in titanium dioxide, the nanoparticles have gone inside the nucleus as well as they are there in the cytoplasm. And same was true with zinc oxide nanoparticles. They are there all over inside the nucleus, as also in the cytoplasm. Some more microphotographs. And one interesting finding in epidermal keratinocytes, we found that this is a control cell, but in the cells exposed to nanoparticles, the nanoparticles actually move from one cell to the other through philopodia. This is quite interesting to see that, why I'm showing this is because one should understand that at the time you're doing observation, one should be very careful and very critical of the observation. So I think this was a very fantastic observation where we can see this, the nanoparticles moving out from one cell to the other. So that also gives you some idea as to how the cells will behave once they are exposed, or once the skin is exposed, how the cells will behave. As I mentioned, it was a complicated process, time consuming as well as very poor statistics. So we looked at another sort of test system, which is flow cytometry. And here I just like to mention that it is a very simple way of understanding. Metry means measurement, cyto means cell, and flow means in a flow. So when you are measuring the cells in a flow, that technique is called flow cytometry. And it is very simple, cost effective, non-invasive, because you don't have to make any blocks to cut the sections and then look at the temp. And it provides very robust statistics because you can measure almost 50,000-60,000 cells per minute. So we gave this hypothesis that if you put them in the flow cytometry, the cell, as the laser is fired, you get a forward scatter, which gives you the size of the cell, and the side scatter gives you the granularity of the, or the density of the cell. So if the nanoparticles are stuck outside the cell, both will increase. If they go inside the cell, side scatter will increase. And if the cells are, if the particles are fluorescent, the side scatter and fluorescence both will increase. So this was the concept that we brought. And this is how we looked at the results, and we found that in human cells, you could actually see that side scatter here is about 6,000, and as we move up in the concentration, it goes to about 60,000, 58,000, almost 10 times. Whereas the forward scatter is not significantly disturbed. So which means that the particles are actually going inside the cell. So just to ensure that everything was fine, we moved ahead. And we also looked at the DNA damaging potential of these nanoparticles. So we looked at the COMET assay, the micronucleus assay, and so forth. So these are some of the tests that were performed with nanoparticles in different cell lines. So as I mentioned to you, that in this case, in zinc oxide, even at lower concentration. So this is another thing which is important. The concentration should be such which are relevant to the exposures. Okay, so we found that even at lower concentrations, we did find significant DNA damage in the COMET assay, and we used 1 millimolar ethyl methane sulfonate as the positive control. These are some representative photographs of the experiments. We also looked at the micronucleus induction in zinc oxide in Fg2 cells, and we found that micronuclei, even at very, very low concentrations, were still significantly higher as compared to the controls. But what was interesting when we looked at titanium dioxide, that at higher concentrations, the titanium dioxide does not get out of the system. It keeps stuck with the cell. So which means that these nanoparticles actually sometimes cover the micronucleus, and sometimes when you're counting it looks as if it is a micronucleus, but in fact these are titanium dioxide nanoparticles. So again, this was a challenge that we faced, and we published this as a sort of photograph or a plate. And then we moved ahead and we also looked at flow cytometry based micronucleus assay, which again provides things in 3D. So therefore there is no interference. So we came up for zinc oxide, we came up with a hypothesis that when these cells are exposed to nanoparticles, the nanoparticles get inside, they cause oxidative stress and lipid peroxidation, damaging the cell membrane. They also get into the nucleus, as I showed you, and it causes DNA damage. They also get into the mitochondria and produce pro-apoptotic factors, and ultimately resulting in cell death. So this paper was published in Toxicology Letters in 2009 January, and I was very pleased to see that in April 2009 itself, within 3 months, it was featured in the Science for Environment Policy of the European Commission in the special issue on Nano Materials. And since then, it has got almost 675 citations, more than that, I think now. It was one of the most cited articles of the journal. So again, it depends on the kind of experiments you plan and how you plan them. And ultimately, these are the things that people are looking for when they are formulating policies. The work on titanium dioxide was extensively cited, and the photographs were published in this book, Sunscreens and Biohazard, published from Canada by Elizabeth Leroux. For ecotoxicity, we had a whole list of species in different sort of systems that we are looking at, right from bacteria all the way up to Drosophila, representing different systems of the ecosystem. But here I will just share with you about Escherichia coli. This was very interesting again. We were looking at the internalization of nanoparticles in bacterial cells. So we looked at flow cytometry and TEM. And for detection, we found that this is the site scatter. And if you look at the control value versus what happens when you have either bare nanoparticles or protein-coated nanoparticles, the site scatter increases drastically, or I would say significantly. Okay, and same was true with titanium dioxide. So which means that these particles actually enter the bacteria. And also when we are looking at the electron microscope in parallel, we found that the bacteria, although it is dividing, the nanoparticles are still there. So which meant that if the nanoparticles are still there, that means that the first generation which is getting exposed, it is carrying forward these nanoparticles into the second generation. So therefore, we looked again at flow cytometry and went up to 3 generations after the first exposure, which was here. And we found that even after third generation, we could find the nanoparticles in the bacterial population. So this was only possible because the life cycle of bacteria is very small, or the time in which it divides is very small. So you can actually do a 3-generation study. Same was true with titanium dioxide. And we confirmed these findings with electron microscopy as well. We also looked at scanning electron microscope, where we could see that when we use zinc oxide, we have zinc and oxygen. But when these particles are coated with protein, such as this one, you find nitrogen, carbon, and oxygen again increased in the SEM, which was very clear. So when you do EDAX, it is very clear that these particles are actually coated. So why I'm saying all this or showing you this, that the more data you provide to the regulator or to the policymaker, the better it is for him or her to, while they are formulating the policy. Same is true for titanium dioxide. So all data that we published. This is another interesting finding, because nanomaterials toxicology or nanotechnology is a very interdisciplinary field. So here was an example where we looked at graphene or graphene oxide for drug delivery. But we found that when you look at graphene oxide and we place our alpha helical protein because of the hydration, the protein structure remains the same. Although in the case of graphene, because of no hydration, the alpha helical protein actually loses the structure. So this is a pure biological problem resolved by a pure computational approach and published in a pure chemistry journal, which is Langmuir. So again, it shows how interdisciplinary this area is. And over the period of time, right from synthesis all the way to in silico prediction, as I've just shown you bits and pieces of these entire repertoire or entire test systems that were there in the IITR that we generated over the years. So I think now we have all the test systems available. Within CSR system, we had another project that was being run with 17 institutions, where IITR was looking at toxicity, all others were looking at various modes of synthesis. In 2008, we did an international conference on nanomaterial toxicology, which was very early on. And we actually got Dr. Sally Tinkle, who had written the National Nanotechnology Initiative of USA. She came and she was a part of the conference. And we were very fortunate that we had very learned people from different countries come and be part of this system. So ultimately what we did, we came up with a declaration, which was to look at what the other countries see should be done at in India. And so therefore these recommendations were then published as part of the overall findings of this conference in CSR newsletter. It is still available as a PDF form. In 2008 it was published as a PDF. So this also gave us a roadmap as to where we should take this forward. So I think it is very important that we don't just do conferences and meetings for the sake of it, but what comes out of it should be recorded. Not recording, but it should be documented and then it should be passed on to the government for various actions. So I think here, since the government is also involved, it would be a good idea to look into how we can utilize the findings of various speakers into coming up with a roadmap for different STI policy for Gujarat. We were fortunate that we got 2 UK RI projects. We also got 2 European Union projects. NanoValid was a mega project with almost 13 million euros as a funding for development of reference methods for hazard identification, risk assessment, and LCA, life cycle assessment of engineered nanomaterials. And we had 19 countries participating, and we were participating from India as IITR. In 2011, we released the first guidance document. During the meeting that was held again in 2011, we did another international meeting on safe use of nanomaterials and workshop on nanomaterial safety. And here we released the guidance for safe handling of nanomaterials. This was the first document released as part of the journal, which had all the publications. During that period, we also trained students and faculty from 8 different countries of Europe, who then went back and started nanomaterial toxicology in their respective countries. So this was another case in point where we were the leaders and we trained people from Europe to go and start doing work. So just to give you a recap as to the speed at which it was done, the initiation happened in November 2005. At that time, I was in Michigan, and the then director of IITR immediately wrote back saying you should start as soon as you come back. So I came back in February. In April 2006, we started setting up facilities. In December, we had submitted a project with NCL Pune. 2007, we had set up the Indian Nanoscience Society. 2008, we had done the first international conference where I think 8 countries had participated to understand what is happening globally. We got 2 projects, as I mentioned, from 2007 to 2012. Another couple of projects we got from European Commission, but CSIR soon realized I think it should also start a network program, and that is how they started a national network program of CSIR institutes from 2012 to 2017. And we did another conference where 15 countries participated in 2011. And that is where I showed you the first guidance document was released. The advanced imaging facilities came up at the institute in 2016, and it was inaugurated by Honorable Minister Dr. Harshvardhan at that point in time. In 2017, we looked at the overall positioning of CSIR for nanomaterial toxicology in Web of Science, and we found we were in the top 5. We were number 4. Number 5 was Harvard. 2017, I was awarded the honorary doctorate for the contributions to nanomaterial toxicology by University of Bradford, and the only person in the history of that university to get an honorary doctorate outside of the UK. 2017, we published our first book on nanotoxicology, experimental and computational perspectives. 2018, CSIR set up a national nanotoxicology facility at IITR. And 2019, the first guidelines for evaluation of nanopharmaceuticals in India came up. It was a joint venture between CDESCO, Department of Biotechnology and ICMR. Later in 2020, a guidelines for evaluation of nano-based agri-inputs and food products also came out. And both these guidelines are free to download from the Department of Biotechnology website. So you can go there and download these websites, these guidelines and read them for yourself. So this is the, and fortunately, I was involved in both these formulation of both these guidelines. Here are the cover pages of the guidelines. And in the meantime, we also looked at other guidance documents that were coming around the world. The first was the one that came out by the Scientific Committee on Consumer Safety, and that was in October 2019. And here, a couple of our references have been mentioned. Another guidelines on the best practices and safe handling came out through the Nanovision Department of Science Technology, Government of India, where the then director of IITR was part of the committee, and our guidance document has been also referenced in this policy. As I mentioned, we published then 3 books in a row, one in 2017, one in 2018, one in 2019, all related to nanomaterials toxicology. I think it's very important that whatever data we generate in the country should find way in the best of journals or the best of books or patents or whatever it is. It should reach the global community. I must thank, I would say, the backbone who were the students, rather, most of them are now part of some university or the other, either faculty or in private sector. And I think they took the challenge along with me, which is remarkable, because I think until you take a risk in your life, rewards don't come easy. And I think I was fortunate that I took the risk, and it was supported by the institution and of course CSIR, which allowed me to do the work that I was able to share with you and reach to a point where the research actually converted into policy. Later, I think
thank you, sir.
TEDx wanted to
a wonderful talk. Now we are open for quick 1 or 2 questions. If any viewer or participants have questions. So we have one question. Sir, can you give some information about the use of BET instrument and its application?
Yeah, so essentially the BET instrument is there. Have you stopped the sharing? Oh, stop share. Okay. Sorry. Yeah, so the BET instrument actually allows you to measure the surface area of these particles. That is the advantage, that you can get a sense of the surface area of these particles. Any other questions? You are Sandhya, you are muted.
Yes, so actually there was a little bit connection issue.
Perhaps there are 2 computers next to each other. That's why there is an echo.
Yes. Yes. So any other questions from participants or viewers? Otherwise, we can move on to next session or the next talk. Okay. Thank you, sir.
Thank you so much.
Now, okay. Now I would like to invite Dr. Bakulesh Khamar, for his talk on evaluation of active immunotherapy for cancer. Dr. Khamar is currently working as Executive Director at the Department of Research and Development at Cadila Pharmaceuticals Limited. He has experience of more than 20 years in the field of preclinical and clinical studies, and also involved in various national and international programs related to the same. Sir has around 90 publications and has led 2 projects. I request Dr. Bakulesh Khamar to please deliver the talk. Please, sir. So you are on mute. Please unmute.
Is it visible now? Screen is visible and
Yes.
audible?
Yes, screen is visible and you're audible.
Okay. So I would like to thank the organizer for organizing this meeting and inviting me to give this talk. So I've chosen this topic because currently in the cancer, immunotherapy is a buzzword, but I mean, it started very long back. As a student of science, we know that we make observation, we make experiment, we do try to analyze and reason it out to get to the facts, concepts, and then we rework. When it comes to the product development, all that scientific knowledge is required to generate some idea leading to a product, and there's in between product development and testing. This product development and testing time is relatively small in the software industry, but is tedious, time consuming, prolonged, and met with failures in the healthcare industry, particularly in the drugs. As you know, today, the immunity is a humoral and cell-mediated, and cell-mediated immunity, which is mainly through the T cell, is responsible for cancer development and eradication. And it got a big jolt when the first product was approved, and in 2013, science considered to be T cell on the attack as a breakthrough of the year, and that's how cancer immunotherapy got momentum. And picture on the right shows that T cell recognizes the cancer cells, and there are so many things in between. So you need an antigen, you need a co-stimulatory molecule, and you need absence of checkpoint inhibitors, which are CTLA4, PD1, PD-L1. I mean, though it is relatively new, upcoming field, the idea is not old. In 1550, in the Egypt, it was noticed that it is possible to have spontaneous remission of the tumor. Then in 2000 AD, it was detected that cancer is an inflammatory lesion, and current understanding is inflammatory lesions are more amenable to immunotherapy. Then somebody detected immune infiltrate into the tumor. Then erysipelas was identified. When you get erysipelas, which is a skin condition, infection by streptococcus, you get remission. You are likely to get regression or remission of the cancer. And in 83, Streptococcus pyogenes was found as a causative organism of erysipelas. And I'm showing this chronology to tell you that observation always preceded development of the science. Though erysipelas was known, it was known that it can regress, but nobody knew what causes it. And then in 1985, surprisingly, not a scientist, but a playwright, Russian playwright, he said there is a relationship between erysipelas and remission of a cancer. In 1896, spontaneous remission following severe influenza was also known. Now, these are all old stories. Currently, immunotherapy is approved for varieties of conditions as listed in the picture on the right side. It includes practically all tumors. Going back, In science, as Louis Pasteur said, in the field of observation, chance favor only the prepared mind. While these observations were made, as mentioned in the previous slides, it was Coley in 1890 who ventured to convert that idea into a product. So he was perplexed how to resolve his patients who are suffering from bone cancer. He gathered information, all the information, analyzed it, and in 1891, he saved the first patient of inoperable cancer. And this is that patient. He had an inoperable tumor in the pharynx, making it impossible for him to swallow. And he got first living Streptococcus pyogenes directly into the tumor by Coley. And first injection did not do anything. So he kept giving those injections. And for 5 months, luckily for us and luckily for both of them, in October, the patient developed fever and the tumor started regressing 2 days back later, and it completely disappeared in 2 weeks. And he remained tumor-free, disease-free, and lived for more than 8 years subsequently. So this was a success story, and then he persisted on those, but the challenge was this was unpredictable, how to give, how much to give, how frequently to give. And if you don't get a fever, you don't get a remission. And death were also seen in the people because it was an infection. He continued elaborating his studies and publishing it. And this is one of the publications which is considered to be groundbreaking. Then after doing lot of trial and errors, in 1898, he came out with a cholecystectin, which comprised of kill organism this time as pyogenes and Serratia marcescens. And the knowledge at that point in time was Serratia marcescens increase the virulence of pyogenes, and that injection induces fever, but it reduces the risk of death. And knowing that now this is feasible and possible, in 1899, Park Davis started producing cholecystectin and selling it. And if you look at the subsequent publications, I mean, it was remarkable that inoperable tumors, he used to get 60% complete responses, which are difficult to get even today. And 5-year survival was more than 75%. The next milestone came in 1908 because everybody was looking with skepticism. But Paul Ehrlich, who got the Nobel Prize in 1908, he also recognized that Coley's observations are right and he can reproduce it. And Paul Ehrlich, because he was a Nobel Prize winner, this got little more impetus to the science. And Paul Ehrlich did another thing. He said that there is a cancer immunosurveillance which is required in the body, which makes tumor grow or tumor disappear. In the same time, the Nobel Prize was shared by McIntosh, who established phagocytosis. So this was a major milestone in the history of immunotherapy of cancer. But if you look back and say why they could achieve this, so Coley had extensively looked at the literature and analyzed that. And after doing that, he did not stop there. He passionately pursued and persisted. And in spite of early failure, he started looking at the successes and started improving his product and improved his success rate. So this is something which we need to learn when it comes to product development for healthcare. Then the next milestone came in 1953 after a prolonged period where Michison said that anti-tumor immunity can be transferred only by the lymph node, not by any other means, whether it's a serum or a blood cells or a lymphocytes. And this was a major milestone. That's something to do with lymphocytes. In between, the cytokines came because there was a lot of improvement in the bioanalytical methods, and there was a recombinant technology developed. So interferons and IL-2 were discovered, and interferon subsequently got approved for hairy cell leukemia and Kaposi sarcoma in 1986 and 88. IL-2 got approved for renal cancer in 92 and melanoma in 98. So all this took away the advancement in the T cell immunity or active immunotherapy. And all the cytokine related products had a minimal improvement, but they were approved. Their improvement were in the range of 5 to 15%. But moving on to the active immunotherapy. So in 1960, cytotoxicity by sensitized lymphocytes was documented, and antigen specific receptors were documented on the cytotoxic T lymphocytes. The major breakthrough came that cytotoxic T lymphocytes will only target one cell, cancer cell or whatever that cell is, and you need to have a T cells for cytotoxicity. And moving quick forward, there are inhibitory molecules. So 1987, the T cell receptor which are inhibitory in nature, CTLA-4 was discovered by Ellison, PD-1 was identified, and PD-1 was identified by Honjo, and he also identified as a negative regulator of immune response. So this puts the brake on the immune system. In 2001, the concept after all this exercise came that there's something called cancer immunoediting, and T cells are required for anti-tumor surveillance. And this is what immune editing means, that if your immune response is strong, your tumor does not grow, it is eliminated, and you are free, as all of us are healthy without cancer. But if your immune response is little low, it reaches an equilibrium state where tumor remains small or grows very slowly. When there's an immune escape, the tumor starts growing fast. And in this all phases, as shown in the bottom pictures, there is a balance from immunostimulant to immunosuppression. If it is balanced in favor of immunostimulant, you have an elimination phase. If you have a balance in favor of immunosuppression, you have an escape phase and cancer keeps growing. And this, looks good, but how does it happen? So while the tumor grows, there is a loss of antigenicity. The tumor cells cannot be recognized. There's a loss of immunogenicity because of the expression of a checkpoints or inhibitory molecules on the surface like PD-1 or CTLA-4. And there's an immunosuppressive microenvironment created within the tumor, around the tumor, which does not allow immune cells to progress or kill the cancer cells. And a picture on the right shows you what happens when you have an excessive amount of checkpoint inhibitors or immunoinhibitory molecules expressed on the T cells or a cancer cells. It leads to exhausted T cells. So you have a T cells present, but they're exhausted and they're not capable of killing the cancer cells. And Schreiber convincingly showed in 2011, which is a science publication, that T cells are key for killing of the cancer cells. And if you have animals which are immunodeficient, tumor grows very fast and animals get killed. But if you have immunocompetent animals, it is difficult to grow the tumor. And this led to the 1918 Nobel Prize shared between two gentlemen, Ellison for CTLA-4 and Honjo for PD-1. But this Nobel Prize paved the way for a next round of immune therapy. Now, what did they do? So from an escape to elimination, if you want to control the tumor and get rid of the tumor, you have to, what they showed is, if you inhibit the negative signal, which is CTLA-4 and PD-1 or PD-L1, the tumor can shrink. You can get remission in the cancer. And they showed first in the animals. This is what Ellison's first publication, CTLA-4 blockade. He used four different types of animal models and showed convincingly that CTLA-4 blockade reduces the tumor and leads to regression. And then that led to discovery of CTLA-4 blockade, which is now available as a EVORI. And phase 3 clinical trial data showed that it improves the survival in cancer. In cancer, it is more important to have a survival rather than tumor regression. Honjo did the same subsequently for PD-1, PD-L1 blockade. He showed that tumor regression is possible and improvement in survival is possible. And this led to development of PD-1 inhibitors. And they first showed it in melanoma. and showed significant improvement. Hazard ratio is 0.42. And then subsequently, there's a plethora of products approved, which includes nivolumab, pembrolizumab, cemiplimab, atezolizumab, avelumab, durvalumab. Either they are PD-1 inhibitor or PD-L1 inhibitor. And then if you look at the picture, they are approved as a first line or second line therapy for varieties of cancer, and this list is increasing. Then comes to our own story of active immunotherapy. So rather than looking at a negative signal, if you try to do something with the positive signal, can you do it? Cancer cells are devoid of antigenicity, and can you do something? And so we developed Mycelic-C, which provides a positive signal, which targets desmocollin 3, expressing tumor cells. And we showed it, it was shown in the animal studies that it regresses tumor and it is improvement in survival. And then it changes the tumor microenvironment. So within the tumor, activated immune cells are increased from 10% to 20%, while immunosuppressive cells within the tumor decreases by almost half, from 47% to 22%. And this led to a clinical trial which approved this product, and it showed that in advanced lung cancer, there's a significant improvement in overall survival. Hazard ratio is 0.64, and when you go to a specific tumor type which expresses desmocollin 3, which is a squamous cell carcinoma, the hazard ratio improves from 0.64 to 0.40. Lower the hazard ratio, better is the outcome. And if you compare that, I mean, it still holds good in spite of all the passive immunotherapy or immune therapy which invigorates T cells compared to the mycelium. The outcome in a squamous cell carcinoma is better with this. With this, I'll stop and invite questions. Thank you very much.
Thank you, sir. We can take one or two quick participants if you have any questions.
Can I ask a question?
Yes, sir. Yes, sir.
Thank you, Kamar sir, for nice talk. I just have one question. How is it in terms of metabolic inhibitors or something? Is it any drug that is coming as a enhancing the immune response within the tumor just by metabolic inhibition and all? Because cellular therapies or these inhibitor therapies are fine, but we know metabolism plays very important role. So is there something related to that, if you have any idea?
Okay, so as far as I know, it is still in a preclinical stage. This is an idea that immune cells within the cancer environment changes their metabolism.
Yes.
And because of the change in metabolism, they acquire immunosuppressive characteristics.
Yes.
And if you change that immune environment or change their metabolic pathway, it is likely that you can invigorate the immune environment and get rid of a cancer. So this is an idea.
But there are few things in clinical trial like that?
No, nothing is in the clinical trial currently. As far as I know, everything is in preclinical stage, because it's difficult to change tumor microenvironment, because it is difficult to penetrate the tumor with the drugs or any other thing. So it is still an idea whose time has not come yet.
Okay, thank you. Thank you.
Okay.
So we have one question from Jaspreet Singh. What is further upcoming development in this domain?
Okay, so in this field, okay, I mean, I could not go into the more details, but what people are trying to do is trying to improve the outcome. So say, for example, PD-L1 inhibitor, though they are fantastic drug, they have improved the outcome to a significant amount, but it works only for a limited number of people. So if you look at it, tumor response rate, they are in the range of 20 to 30%. So, and the reason are, I mean, multifold. So people are trying to evaluate combination therapy and trying to combine them with other therapies, or trying to identify new targets and develop immunotherapies around it. But so far, success has eluded all of us. So there are no success in that area. The other area which is there is, you take the immune cells from the tumor of a person, revigorate and transfer it back, either as they are or by improving using biological methods or modifying that gene. And this is also getting momentum. So one part of it, which is known as a CAR T cell therapy. So CAR T cell therapy is currently approved for leukemias of certain kind. So where you take the cells, change it and modify it genetically and send it back. And this is based on the concept that one T cell kills one cancer cell, and then it has to, so at the same time. And cancer, when they are a significant size, they are more in numbers compared to the lymphocytes available in the body. So how do we expand more? So they are trying to expand more and send it back into the system in a targeted way, so that one CTL will match the one cancer cell, and then they can kill almost all the cancer cells. So that's the concept.
Okay, sir. Any more questions? If there are no more questions, we can take a quick break of few minutes and then resume. So I think we can resume for the next talk at 11:45. Thank you, sir.
Thank you. Thank you, everybody.
Welcome back all. I would now like to invite Dr. Subir Majumdar, Professor NIAB, Hyderabad, to deliver talk on biotechnological interventions in animal health affecting human welfare. Dr. Subir Majumdar did his PhD from National Institute of Health and Family Welfare, New Delhi. He did his postdoctoral research from School of Medicine, Southern Illinois University, Carbondale, USA, and Department of Cell Biology and Physiology, University of Pittsburgh, USA. Previously, Sir has worked at National Institute of Immunology, New Delhi. Currently, as already mentioned, he is distinguished professor at National Institute of Animal Biotechnology, Hyderabad. He is fellow of National Academy of Sciences, Indian National Science Academy, and Indian Academy of Sciences. Sir has more than 50 publications in various reputed peer-reviewed journals to his credit. I request Sir to please deliver the talk. Please, Sir.
So please allow me to share the slides.
Yes, sir.
Can you see my slides?
Yes, sir.
You can?
Yes, sir. Yes.
Okay.
Yes,
So good morning to all of you, and thank you to the organizers for inviting me here. And I will try to finish my talk in 20 minutes. So thanks to all the colleagues and friends who are participating. So I'll be talking about biotechnological interventions in animal health affecting human welfare. I work at National Institute of Animal Biotechnology, a DBT institute at Hyderabad. So to address this, I think 20 minutes is not enough to say everything, but I will just give hints and ideas and varieties where we should be focusing, giving few examples, not everything. So the key areas of research for this particular field where biotechnological interventions can be done for addressing animal health is I would begin first with animal nutrition, then infectious disease. Of course, in nutrition, dietary supplements with nanoparticle deliveries, biotechnology has a big role because everything is not absorbed by the large intestine efficiently. Infectious diseases, I think development of good diagnostics, development of effective vaccines, long-term effective vaccines, and therapeutic molecules, etc. Animal reproduction is a very important part because reproductive health is an important part of human as well as animals both. And we have to overcome infertility in several animals. And I'll come to that. Certain diseases affect the fertility of animals. Definitely, we have to work with the transgenic technology. With the advent of knowledge of various genes and their functions in health, we can generate disease-resistant animals. And there it comes that if we can do animal genetics and genomics work, where we can develop elite herds of animals who are immunologically more efficient to protect themselves from the organisms, then that would be a better way of working. And for this, as well as the gut microbiome studies, as gut microbiome nowadays is said to affect metabolism and several functions of body, I think for all the three, it is very important to have a very good bioinformatics setup, because without that, we really can't address several issues. So biotechnology in a nutshell in this area is what it is. Now I will just give you a glimpse of what I can say in a few minutes. So as I said, most important is livestock genomics, so that you can choose and select and breed elite herds which are immunologically really elite, and they can have several kinds of diseases just avoided because of their quality. So I will talk to it when I come to it. Livestock zoonosis has become a major problem where the diseases are actually carried from the animal to human. We are seeing at present the COVID-19, and there we have to focus on epidemiology, pathogenomics, diagnosis and prevention. Their biotechnology can work on pathogenomics, diagnosis and prevention. And our institute's motto is animal health for human welfare. So I think that needs to be correctly addressed. For that, I think it is very important that we detect the disease at right time and intervene them. So you need to use biotechnology to quickly detect the disease, just contain them where they are and not allow them to spread, and also develop vaccines. But few of the disease which kits are very important for us in India is CHF, African swine fever, leptospirosis, Toxoplasma gondii, Brucellosis, Japanese encephalitis, Theileria. In addition to such detection of disease in the kids, we should also address quick detection of antibiotics in the milk and also detection of pesticides in the cattle feed, because pesticides are affecting our cattle and they give milk that is consumed by human, that also affects the human health. So needless to say that as we are staying very close in contact with the animals, antimicrobial resistance has become a big challenge, because if these animals are treated with various kinds of antibiotic repeatedly without proper knowledge, they generate bugs which come from them to human, and it's a big trouble and problem in developing countries that we have to keep in mind. So I'll take one example of brucellosis. So according to WHO fact sheet, although approximately 500,000 brucellosis cases are reported annually, but the true incidence is always 10 to 25 times higher than the reported number of cases, because everything cannot be detected. And you see how many various kind of brucella can affect various organisms. And if it comes to human, it causes infertility and febrile illness, and that is very, very risky for human. So in livestock, it is frequently causing chronic disease with reproductive failures that contribute to production losses. And in humans, it causes an often chronic febrile illness and infertility that is frequently underdiagnosed. So it is important to correctly detect brucellosis before treating. So presently, whatever assays are there to detect brucellosis is by the superficial layer of the membrane, that is LPS, and you have to detect it. So if the animals are immunized with the vaccine, and if the animal is naturally infected, you cannot differentiate between them. So recently, in our institute only, a new approach has been taken, that instead of the surface protein, we have done the intracellular events in the brucella and found that BMP5 is a protein which is secreted in the blood of the host. So if we can detect this protein and develop ELISA, as well as we have developed lateral flow assay, you can really say that this animal is not vaccinated, but it was either exposed or having brucella. It's a wonderful technique where the biotechnology is needed to generate new detection kits where you can really dissociate the one which is vaccinated by one which is not vaccinated. But for that, you have to really study the cell biology of the microbes and pick up a protein which is secreted in the blood so that you can detect it. So there are very good studies now, brucellosis in India, comparing exposure amongst veterinarians and paravets and animal handlers. Very recent paper by Dr. Dhan and Gadava group. Wonderful thing to go through, and you will find that how important it is to address this brucellosis. The other issue is mastitis. All of us know. So there is nothing to check or early diagnosis of mastitis. That means from normal, when it is early mastitis, is there any way to detect completely confirmed way that there is mastitis? And if it is diagnosed and treated well at this point of time, the not only milk production gets doubled, but you get rid of developing a lot of resistant bacteria. So I will show you what I mean by this. The characterization of Staphylococcus aureus isolated from milk samples of dairy cows in smallholder farms in Ethiopia. You see, every country has done. This is in Chinese herd, and problem is of antimicrobial resistance of Staphylococcus aureus isolated from bovine mastitis. So mastitis is giving trouble to the cow. That's one thing, but it is generating a lot of antimicrobial resistant Staphylococcus aureus, which through milk can go to human and cause a havoc in human beings and affect human health. So there's an urgent need for improvements in the antimicrobial stewardship and infection control programs to reduce emergence of AMR in general. It is necessary to detect the infection at preclinical stage of mastitis so that you can control this infection to reduce the emergence of AMR. So the Oxide subclinical test is very, very important for mastitis. And recently we have developed a kit where you have to simply use the iron nanoparticles and have the buffers which are appropriate, so that if there is even a small amount of bacteria, it will bind and it will really make a clot. So if you put on a filter of a membrane, you will find it. So this is better, nicely shown in the next slide. And you just take 10, 12 drops of the milk, put it, and step 2 by magnetic isolation. You can just get them clogged, wash, and just invert it on this tissue paper or absorbent sheet. You will find this is negative, and one which really shows you the clustering is positive. So this is portable, stable at room temperature, and farmer friendly. You detect the disease ahead of time so that you can treat it with mild antibiotics and you don't have to really go for any bigger antibiotic treatment. One of the other important aspect is some of the commonly used pesticides in India, like chlorpyrifos, atrazine, endosulfan, etc., are known to exhibit endocrine disrupting effects in animals and humans. So studies has reported the presence of such pesticides and their metabolites in the milk and meat of the farm animal. That's dangerous because we can take any grain, rice or wheat or anything, and we will just use it by polishing it and doing so many things. So pesticides are gone. But our livestock eats the remnant, which is never washed to give them, and they do ingest, and it can come in the products of theirs, and it can cause to human beings endocrine metabolic dysfunction, cancer, infertility, subfertility. These are all reported information. So now it also causes another problem, that exposure to endocrine disrupting chemicals can induce epigenetic changes in the germline, which are then passed on to the progeny. So basically it is going transgenerational and via the modified germ cells. So they may induce changes in DNA methylation. So you can see these are the normal sperm. This exposure can really do changes, epigenetic changes, and which results into poor semen quality and failure to fertilize ovum, actually affect the health of the animals. And we can see the detrimental epigenetic changes inherited by the calf, whose parents are exposed to such kind of disruptors. So we are trying to develop, and we must develop certain enzymes or chemicals or something with which we should treat the feed and fodder before, to really change the endocrine disruptor into some other chemicals which are not harmful to the body, or somehow restrict the usage of these endocrine disruptors in the field. We also know that there are several incidents and events where the whole world gets threatened by the breakouts of certain diseases in the poultry, because that is most commonly used meat. And this is just a figure that how the animals are culled, eggs are thrown, and in a country like ours, which is a developing country, it's very difficult for a marginal livestock keeper to really tolerate this and bear this. So there are biotechnological interventions, and long ago, I think in Roslin Institute, they started to generate transgenic chicken expressing RNAi against the virus so that the virus cannot replicate even if it has infected the chicken. And there are a lot other studies where avian influenza, this is the report of avian influenza, where viral mRNA is really interacted with the siRNA and breaks it down, limiting its replication. Very recently, the Mareks disease causing virus, which is by herpes virus, and they have shown that they can really do generation of a transgenic chicken, where in vivo, they have shown that this is inhibited, their growth is inhibited by expressing Cas9 and gRNA against ICP4 protein. And what is this? Is TOL2 transposon construct encoding Cas9 and guide RNAs specific to immediate early infected cell polypeptide ICP4 of MDV. So it doesn't allow the virus to be formed inside the cell. And they found that when they challenged such animals with the virus isolation was significantly lower in the transgenic chicken as compared to either a control or only Cas9 inserted chicken. So this is a very good method and very nice way to generate transgenic animals. This is an example of transgenic poultry, but you can generate any kind of transgenic animals using biotechnology to really reach what you want to do. Now, as I said, that it is very, very important to detect the disease at early time point. So let us say SARS-CoV-2, and today it takes 4 hours to really give you a result, and actually results come next day or late in the evening. But what we need is a fast detection of the disease or the organisms. And one of the ways which is worked out in our place only is that to develop a gold nanoparticle, we have developed gold nanoparticle based electrodes where antibody is really tagged on it, and there is an electric current flowing with gold nanoparticle and antibody, and you can really watch the flow of the current. As soon as spike antigen of the SARS-CoV-2 is bound to this antibody, the resistance changes. It becomes more resistant, and you will find that there is a change in the resistance pattern, and then you can make a cutoff that presence of spike antigen is there or not. And you can see very good result of here that you can see as compared to control. This is the current which is coming. It's sharply gone down in case of SARS-CoV-2. But when you use other viral proteins, MERS and others, you find that there is no change. So it is very, very specific to SARS-CoV-2. So you can develop this. The beauty of such things that antigen can be detected within few minutes instead of it taking hours. Now, I was talking about the livestock genomics, that how that can help. So we have developed a first chip of the country, which is the biggest of the world also, because we have around 11,496 more SNPs than the 777K SNP of the Epimetrics, which is developed in the Western world. So this is specifically for the Indian cows. And we have used all, at least we used 43 varieties of cows to develop this chip. But now we can go in the field and really use this chip. We named as Indigo, and we can develop patterns. We can really find animals which are resistant to certain diseases, and we can find animals which are prone to certain diseases that we can get in the field. And when we use this SNP chip, it can help in selecting disease-resistant herd, and we can generate a healthy herd. So after doing this, we can select which germline is really very, very good, immunoprotective, for example, and which are the graded animals which are not good and which are admixture, and we can throw them out, and we can keep the pure breed which are immunologically very competitive and breed healthy animals so that they do not fall to the microbes. They remain healthy, they give more milk or meat, and they kind of disease to human, and they are not prone to, or they are not amenable to, or there is no requirement of treating them with antibiotics because they are healthy, decreasing the chances of developing antimicrobial resistant or AMR in them and preventing human health indirectly. I'll come to other issue, which is, if you go to the field, you will find so many human therapeutic proteins available, but they are extremely costly. You can see the cost here, and poor people cannot. Certain things are very, very costly. And I know for that one vial of gamma interferon costs around 14,000 rupees, and one full treatment can take around 3.5 lakhs for 6 months. So this is not affordable by masses. In addition to this, we also need a lot of FSH, LH for inducing fertility in large animals, which is cattle and buffaloes. It is not possible to buy them. It's very, very difficult. So if you can generate these therapeutic proteins in the milk of an animal using biotechnology, that will be a great help for the country. So we, in this direction, we have developed this. We have isolated buffalo beta casein promoter and characterized it and tagged human gamma interferon gene. And the idea was that you take that gene and tag to the human gamma interferon and really make a transgenic animal and develop that animal in the milk of that animal along with its own proteins. This protein of human origin can also be generated, and then the cost of 13,000 very costly vial can be sold at a very cheaper cost. Now, while doing this, we have also found that generating a transgenic animal may be a problem because there are a lot of issues related to GMO at today's date in our country. So we have started doing one more biotechnological intervention or innovation, that rather than making the whole animal and coming into the line of criticism, what we have done, that we have started directly injecting the gene of our interest at a growing udder. So that time of growth of udder you have to find out for each species, where this gene gets integrated while the cells are dividing. And then whenever the milk production occurs, because there is a beta casein promoter, and it secretes it should ideally secrete in the milk. That work we have done as a proof of principle in mice and rabbit. And this is how we inject in the udder gland, and it flows in the udder gland, this solution with containing the suspension of the DNA. And we have shown that if you take a normal udder gland, there is no fluorescence. And if we tag it with GFP, only the transgenic udder glands give you GFP. We have also shown by Western blot analysis that we can express human gamma interferon. And we have also shown that if we do kind of the whole body, various organs analysis, we find that only udder gland is expressing the protein of interest, what we have injected or into the udder gland, but no other part of the body, brain, liver, kidney, doesn't express that, showing it is very specific. Then we have ELISA to show the human interferon gamma kit was taken from the market, and we have shown that we can generate in the milk as well as lysate of the udder gland of the mice, and also the same work has been repeated in rabbit, and it is successfully give us production of human therapeutic proteins in the milk. Now, DBT has given us a big grant to go ahead and work in the large animals. The other aspect of today's challenging aspect is major outbreaks of India, Southeast Asia, that how they have problem is that if you see this figure, you will come to know that diseases are coming from animal to human, coming from human to human, and then from going from human to animals, what we call as reverse zoonosis, and definitely coming from animal to animal also. It's too complicated. And to resolve this, it is very, very important that we organize ourselves and try to do justice, that how this can be quickly addressed, because COVID-19 has given us a lot of ways to understand. And this is what doing everything together is nowadays called as One Health. There are slides of One Health I'm not showing because lack of time, but we have to understand that if we take care of the health of everyone, we are fine. But here, I would emphasize you to see what is that One Health. One Health means livestock animals. One Health means wild animals also. One Health means human beings also. So it is extremely important to get the people who are working with the human beings, that is medical doctors who are giving treatment to human beings, veterinarians who are working with livestock and pet animals in the cities and villages, and in the wild, there are health workers who are serving to the cause of the wildlife health. So you need to bring all these three partners on one platform and create a consortium or a situation where they learn to shake hands with each other. There are common websites where they put their disease outbreaks or first appearance of any disease. They can see and interact with each other. They become friendly and they have phone numbers, they have addresses, emails, and they can interact and they can together address this issue. Otherwise, they are all working in isolation. So with the great understanding of the DBT, they realized, and they really now has awarded us a very big grant called as One Health Center, which is established at NIAP, and the grant name is One Health Consortium for One Health. And the objectives are to establish a network of laboratories at centralized and field levels to estimate prevalence and burden of select diseases through serological and other methods. So biotechnology will help in developing these methods, which are quick and cheaper, to detect pathogens where needed by serological or molecular tests, to model data for disease forecasting and assess the risk. We have taken Nivedi from Bangalore for this work. Dr. Suresh is working with us to generate tools and reagents for emerging zoonotic diseases. So at present, we have taken select 10 diseases because the main criteria is to bring on one platform all the people who are involved in this work. Few diseases are taken as kind of a source of beginning how to work. We have also taken transboundary diseases because our borders are giving from west we get CCHF from Pakistan, northeast we get SF, PRRS, Nipah, swine influenza. So we have to be very careful that we cover our diseases which are predominant in border. Also, this is the small set of diseases we have taken, but what's most importantly done that we have taken AIMS Delhi, AIMS Jodhpur, ICMR, RMRC, Gorakhpur, Dibrugarh, Nazareth Hospital, Shillong, Gandhi Medical College, MGR University. So these are all the medical centers or people. And then from veterinary side, we have taken IVRA Bareilly, Gadwasu. Each of them have got few states to cover for epidemiological studies and surveillance studies, MAFSU, NIAB, and Tanuwas. And on the northeast, there is a Assam Veterinary College at Kanapada, as well as several of other ICR centers. So they will work on the veterinary science, they will work on the patients coming to hospitals. And if some patient is detected positive, they can go to their home also and find out whether other members are affected. NRC Meet will help us with foodborne diseases, and Nivedi will help us with statistical planning of design of the experiment, sample numbers, and also disease forecasting. So with this, I will end that we think always that they gave us the diseases and they think that we gave them the diseases. So we have to work for each other. And what is the motto of my institute is animal health for human welfare. It fits here. So though the problem is big and ground is vast, but human beings need to collate all their knowledge, including biology, machinery, artificial intelligence, that is the biotechnology, for detection, prevention, and curtailing health effects of zoonotic and reverse zoonotic diseases, inclusive of human and animal health, taking doctors, vets, environmentalists, and plants and soil health scientists together on one platform. I think we can solve this issue. With this, I will close my presentation. Thank you.
We can quickly take one or two questions.
Sir, actually, I want to ask that why particularly that zinc oxide Fe nanoparticles are binds to bacterial cells.
Why they want? No. This is, you see, they have some pH changes in the buffer, and
Okay.
that's the trick of the trade. He is going to patent it. Pankaj Suman is the scientist who works on this. If you want, I can connect you to him.
Okay, sir. Okay. Thank you.
Sure.
So we have one question. We have one question from Jaspreet Singh. Is there any machine learning model that is being used currently?
We are developing machine learning tool for our genomics research, and it's expensive. For the first tool, they have taken 22 lakhs just to give us a proof of concept, whereby just by inserting the DNA of any animal, just directly you can get the result that whatever is our phenotypic character. So you have to use large number of animals, and then you have to teach the machine that if this kind of SNPs come, this is either immune dominant, or for example, more milk giving, or for example, any criteria you want to take, heat resistance or anything. So we are beginning. I think we are not there at this point.
Okay, sir. Any more questions from the participants? So, thank you, sir.
Thank you.
It was wonderful talk.
Sure.
Now we have small change in the schedule. Unfortunately, Dr. KG Tirumuragan will not be able to join us, but instead we have Dr. Subhash Jake sir with us. I would welcome and would like to invite Dr. Subhash Jake sir, Assistant Professor, Anand Agriculture University, to deliver a talk on metagenomics in disease diagnosis. Dr. Subhash is working as Assistant Professor at Anand Agricultural University and specialized in the field of animal biotechnology. He has more than 60 articles to his credit and in various reputed journals. Please, sir.
Thank you. Thank you, Dr. Sanjay, for nice introduction. So, it will be very hard and a mammoth task to describe about metagenomics in disease diagnosis in just 20 minutes, but I will try to cover the topic in brief. And it's also very hard to fill in for the speaker who is actually going to deliver the lecture. He is a very renowned personality in field of disease diagnosis. I am just a beginner, so please bear with me. And let me share my screen. Am I audible currently?
Yes, sir.
Okay.
Yes, you're audible.
Okay, so as we are already familiar that disease diagnosis is a very complicated process and it requires application of different approaches and different tools. So metagenomics is one such approach where we can try to identify unknown pathogen from an environmental sample or infected or disease sample. So I will be covering about basically starting with diagnostic microbiology. What are the limitations? Why we need to go to metagenomics as a diagnostic tool? A brief overview about high throughput sequencing technologies, and then I will share my own experience about how we can employ this tool into diagnosis of a particular disease. So diagnostic microbiology is like traditionally it was done using microscopes, like Robert Koch, who had diagnosed the first and observed first animalcules under microscope. So it is like simple observation of microbes under microscope and to detect them in various specimens. But in this case, like whenever we detect more than one microorganisms, it is described as contamination in case of traditional microbiology. So we need to come up with approaches where more than one microbe is infecting the individual. So these are different approaches where diagnostic microbiology is employed. First one is molecular diagnostics, which we will go in detail today. Then microscopy, which is cumbersome or unrewarding. And in many cases, culturing of the microbes is not possible because they are present in tight symbiosis with one another. Another method is immunoassays and detection of nucleic acid sequences, or you can use 16S rRNA gene sequence for the identification of microbes. But in this case, this will only tell you about the identification, what the microbe is. It will not tell about the pathogenicity the microbe presents for a certain individual. And also for viruses, no such universal tool is available where we can diagnose the specific pathogen from a sample. Also, in case of microbes, they are very notorious. They like to play with their genomes and with their genetic information. Like here you can see this H7N9 virus. It contains different genomic segments from different viruses, like it contains segments from domestic ducks, wild birds, and domestic poultry. So all different segments are combined from these different species, and they present a novel virus. So that is that results due to genetic assortment, and that virus will provide a new opportunity for infection to different species. So in case of microbes, we need some high throughput and rapid diagnostic tools which can diagnose such kind of changes in real time. Also, in case of microbiology, we cannot culture most of the microbes, and that is termed as great plate count anomaly. Like when we observe the microbes under microscope and when we culture them, we will find that only less than 1% of microbes are culturable, which are observable under microscopes. And this fraction is also so much lower in case of aquatic environments. So traditional microbiology cannot detect or diagnose the infections which have been resulted from unculturable microbes. Also, the microbial diversity has no limit. Means if you want to detect a microbe or microbial infection, so it is not possible that we can only rely on one particular approach. So here there is one interesting study which was conducted by Donachi and co-workers in 2007. What they did, they collected 7 different metagenomes from different habitats, different environmental habitats, and they tried to catalog the microbial diversity with the help of molecular methods as well as culture-based diversity methods. And so if one argue that molecular methods are superior to traditional method, then it should happen that whatever microbes we have detected with the help of culturable methods, they should be represented into molecular methods. But this was not the case. You can see here in this case, one habitat, the microbes which were detected by molecular as well as culturable methods, they were entirely different. There was not overlapping of a single microbe at all. So you can see only one method will not be sufficient. We will need a combination of approaches for this microbial disease diagnosis. Also, the number of species is, as a biologist, one is always interested in what is the number of, or what are the number of species which are present on the earth. And there are no means specific numbers are available. So it is very much useful to catalog the diversity which is available in case of microbes. And there is one quote which is given, like, once the diversity of microbial world is cataloged, it will make the astronomy look like a beautiful science. So you can see that there is so much yet to be discovered in case of microbes. These are some of the numbers which have been given for identified modern species. So mostly you can see whatever have been sequenced or identified, they includes animals or invertebrates or plants. But in case of microbes, there are very less information is available. So there is very further scope to improve this identification of microbes using sequencing methodologies. Another useful scenario where this sequencing is useful, you can see this is for diagnosis, we are interested in identifying SNPs or single nucleotide polymorphism, because these SNPs are responsible for, in many cases, they are responsible for genetic diseases. Like here you see one example, simple one nucleotide change results into change of amino acid from glutamine into valine, and this results into sickle cell anemia. So you can see how a single nucleotide change is responsible for such a devastating disease. So that's why we are also interested in SNPs with the help of sequencing technologies. So here I have included two different graphs, which shows one graph shows the trend of sequencing costs over the time. You can see as in 2008, the sequencing cost has drastically reduced. Means it was initially suggested that sequencing cost will follow the Moore's law. So as the chip's sequencing capacity will double, the sequencing cost will decrease. But in 2008, suddenly this fall in sequencing cost has outpaced the Moore's law. That is due to the advent of next generation sequencing technologies. And also you can see another example here. The trend of different searches or different projects with this word microbiota and microbiome is presented with help of Google Trends. So you can see after the advent of this DNA sequencing technology, there is huge increase in the people or search which is related to microbiota or microbiomes. So what are these next generation sequencing technology? They are also known as next generation sequencing technology or massively parallel sequencing technology, high throughput genome sequencing technology. So these are all like different names for a single next generation sequencing technology. But in nutshell, these are the technologies which produces millions of nucleotide based sequences in a short time. So initially what we were doing sequencing with the help of Sanger sequencing. And throughput of that technology was very low. So as compared to that Sanger sequencing, this next generation sequencing technologies can produce millions of nucleotide based sequences in a short time. So this is the article which shows in 2008, first complete genome of individual was performed. The individual was James Watson. So this was performed using 454 sequencer or pyrosequencing technology. And you can see as compared to Human Genome Project, which almost taken 13 years to complete. This was performed in a very rapid or very short frame of time. And later on, 454 sequencer was introduced by Jonathan Rothberg for wide commercial use. So these are the different NGS platforms which are available for sequencing. So this is GSFLX or Pyro sequencer, which was available from Roche. It is currently outdated now. Then this is genome analyzer from Illumina, then Solid. This is also outdated. Ion Torrent. So this is the field where tremendous competition is available, and so whatever is lacking in progress or development, it is falling out of the competition. So that's why this 454 sequencing and solid applied biosystems both have been outdated due to high sequencing cost and low throughput. Recently, this sequel from PacBio is also introduced, which has very long read length, so you can sequence entire microbe in a single stretch with this machine. And now it has also become very much portable. So like Minion or pen drive like USB device is available where you can load your sample and you can also perform DNA sequencing in the field. So these NGS technologies have different applications like discovery of new microbes by metagenomic approaches, then investigation of microbial communities, analysis of viral genomes, and detection of low abundance antiviral drug resistance mutations in patients. So it is very much useful if you consider healthcare scenario or disease detection processes. These are some other applications of next generation sequencing, like it can also be used for de novo sequencing if you want to sequence anything new, microbe or genetic or plant, animals, whatever, bacteria. You can also use this for resequencing or confirmation of already existing mutations or SNPs in different individuals. Then you can also use this for transcriptome analysis, that is for detection of expression of different genes or global transcription profile across different samples like drug treated samples or other kind of control and treatment samples. Then you can also use this for gene regulation studies, epigenetic changes, that is DNA methylation patterns, then metagenomes and microbial diversity, as well as you can also study ancient DNA, which are recovered from fossils and many old samples. So what is metagenomics? So metagenomics is basically the study of DNA of the uncultured microorganisms. As we have already seen that 99% of microbes cannot be cultured. So you can study rest of the microorganisms with help of genome sequencing or DNA sequencing. So in case of a genome is entire genetic information for a single organism, while in case of metagenome, what it is a collection of all the genomes which are present in a particular environmental sample. So it is basically a community of organisms or collection of genomes from a community. So with metagenomics, we want to try to answer the basic questions like who is out there, means what kind of microorganisms are present in particular habitat and what are they doing, so what kind of functions they are exhibiting, and in a community what role they are playing and in what proportions they are present. And also with all the metagenomics, you can compare different samples between different habitats, so that will give you a larger picture about how these microbial communities are working in an environment. So this is metagenomics pipeline briefly. So for this, you have to collect the samples from environmental habitat. Then you have to just extract the nucleic acid, whatever DNA or RNA, or you can extract both. Then you can go for shotgun metagenomics. So shotgun metagenomics is the approach where you sequence entire DNA which is present in a sample without any selection or without any bias. And then another approach is amplicon sequencing. So here you can sequence selected portion like 16S or 18S or some gene specific regions, they are amplified using primers, specific primers, and then they are sequenced using next generation sequencing technologies. So after this library is constructed using these valid molecules which can be sequenced using NGS technologies, then NGS is performed. So after NGS, we obtain sequencing reads, and those sequencing reads are initially checked for quality checking and filtering. So whatever bad reads or chimeric reads are there, they are removed. Then quality filtered reads, the reads with high quality sequences or bases, they are aligned against reference sequences, or if you are performing de novo sequences, then you can go for gene prediction. So basically, at this step, you want to try and identify what are these sequences for, what these sequences codes for. So you align them against reference sequences, and you go for taxonomic or functional annotation, and that will give you non-pathogen. Otherwise, you can go for de novo assembly and then taxonomic and functional annotation, and you will come to know about any novel pathogen if it is present in your sample. So this is one of the case study in which we have employed the metagenomics for diagnosis of infectious diseases. So as you are aware that poultry industry is very much is present in very high numbers in case of Gujarat and also India, that provides a number of eggs and meat chicken samples to the community. So on one farm which is present in Sarsa village, the outbreak was reported that there was death of several birds at a single time simultaneously, and the birds have shown the symptoms like difficulty in breathing, swelling of the head. There was continuous nasal discharge, sticky nasal discharge, the reduced feed intake, and cyanotic comb. So comb were cyanotic and red eye and dull, and mostly the birds were dull and they preferred to lie down as compared to healthy birds. So they contacted this veterinary college that what is if to identify the cause of this disease. And these are the effects of this outbreak. So the disease was with slow spread. It took 3 to 4 weeks. It has high mortality with rapid production drop, and recovery taking too long, 6 to 8 weeks in recovery. These are the post-mortem lesions. So after, at the time of necropsy, we found that there was swelling in subaqueous fossa, and there is presence of fibrinocaseative material in our infraorbital sinus. So these post-mortem lesions revealed that there should be some kind of respiratory disease. So with that intention, we collected tracheal aspirates from the infected samples. So tracheal aspirates were collected from 6 birds showing respiratory symptoms. Then these aspirates were pooled in group of 3, and they were processed for RNA extraction using QIAM viral RNA mini kit. Then library preparation and shotgun sequencing on iron torrent machine was performed. So when we obtained RNA reads using RNA sequencing, these reads were filtered against poultry genome. So the idea was to remove the reads which were originated from host or originated from poultry. And then reads were subjected to blast scan against database containing all bacterial sequences as well as database containing viral sequences of NCBI, including sequences available at NCBI flu database. So after this, whatever sequences we have obtained, we tried to annotate those sequences. And then again, after identification of specific pathogen, we tried to virus isolation and sequencing. So these tracheal aspirates and swabs which were collected, they were filtered with 0.22 micron filter to remove all other particles other than viruses. Then they were inoculated in allantoic cavities of specific pathogen-free eggs. So the specific pathogen-free eggs are widely used in diagnosis. They are free from any kind of microbial infection or organism. Then allantoic fluid was isolated, harvested, and viral RNA was isolated from that, and those were sequenced using next generation sequencing technologies. So after obtaining of this sequencing reads, they were quality filtered by Prinsic using this criteria, and they were assembled and using the reference genome of H9N2 strain, because this was the strain which we obtained in the metagenomic sequencing, which was earlier performed. And then nucleotide alignment was performed using muscle algorithm with against H9N2 virus strains, which were isolated from India, and also all available Asian strains and 3 different H9N2 virus lineages, which were available at that time. So after this analysis, so these are the results. Like initially, wherever we sequencing was performed in 3 different groups of metagenomes, we found that majority of the reads were matching with the host genome. So this is the main drawback of this technology, means you have to filter out whatever host genome is there, otherwise you will not get your useful reads. After filtering of this host genome, when we compared this metagenome by blast analysis, we found that this avian influenza virus was coming as a prominent hit in all of 3 metagenomes. So that gives some idea that the poultry flock was suffering from avian influenza. And then the virus isolation and sequencing was performed. And when those sequences were compared or aligned against different avian influenza viruses like H1N1, H2N2, H3NT, which were commonly affecting the chicken populations, we found that the majority of identity was against H9N2 viruses. So that gave the causative agent that the chicken or poultry flock was suffering from H9N2 infection. And in case of mapping of virus isolation with another tool also reported the same thing, maximum mapping against H9N2 viruses. So this also shows the difference between vaccine and SARS isolates. So there is very less nucleotide changes, but still there were some nucleotide changes which were responsible for vaccine failure and causation of disease. Then we performed glycosylation site analysis. So this glycosylation sites are very much important in virus binding and then virus entering into the cells. And we found that in case of HA gene, this segment for its HA gene, we found there is change in glycosylation site between isolate, which was the sequence, as well as vaccine virus. So that might be responsible for this infection of this chicken. We also performed epitope analysis. In that also we found several different epitopes which were differing between SARS vaccine virus as well as field isolates. So basically the sequencing has revealed that there were some SNPs or some nucleotide changes which have resulted into increased pathogenicity of field isolate, and that's why it has overcome the vaccine virus. Detailed analysis of this different receptor binding site, cleavage site, and hemagglutinin binding site, those were also performed, and we found that the isolates which were sequenced, they were differing at various different locations from reference viruses or vaccine viruses. So those mutations were responsible for evade of the immunity. We also tried to find out the substitutions which are favoring the human infection or poultry infection. And we found that at several places, the nucleotide changes were favored by poultry infection. So that resulted into localization of these isolated chickens. Then we also perform phylogenetic analysis using whole genome sequences of all these viruses. So we obtained whatever different avian influenza viruses were present in NCBI databases. When we compared these different genes, we found that the isolate which was sequenced in our study was mostly conforming to the G1-like lineage. In case of HA gene, in case of NA gene also it was confirming like G1-like lineage. So there are different lineages are available. Majorly there were 3 different lineages. So we have included all 3 different lineages in analysis. So in case of energy, also we found that it was conforming to G1 lineage. And rest of the segments were also analyzed using this phylogenetic analysis. And we found that it is basically like a genetic assortment like event where different segments were coming from different viruses. That's why the virus was very challenging to contain this virus with the help of available vaccine. So with this, I want to conclude that this NGS technology is very much useful. Like in case of current scenario, also you can every day you will read that new mutation is coming up in case of SARS-CoV-2 genome. Recently, we have seen that Omicron variant is also coming. So with the help of this NGS sequencing technology, what we can do, we can rapidly identify a genetic mutation or genetic change which is present in pathogen in case of different diseases. So this is very much useful in case of disease diagnosis. So metagenomics is a very useful tool. As you can see in our study also, we found that with the help of metagenomics, we were able to diagnose this disease which was occurring in poultry in very short span of time, like within a day or 2. So with this, I want to conclude my talk. Thank you. You are muted, Dr. Sandhya.
Thank you, sir. I thank you on behalf of GVS for sparing your time and delivering talk on such a short notice. Thank you so much, sir. We would quickly take 1 or 2 questions from the viewers or participants. If there are no questions, we can move to the next talk. Thank you, sir.
Thank you very much.
Okay, so for the next talk, I would like to invite Dr. Mangal Singh Rathore to deliver a talk on genetic engineering for abiotic stress tolerance using plant genetic resources of saline habitat. Dr. Rathore is presently working as principal scientist at CSM CRI Bhavnagar, that is Central Salt and Marine Chemical Research Institute. Sir has research experience in fields of plant biotechnology, marine biotechnology, and ecology. The area of expertise revolves around in vitro regeneration, somatic embryogenesis, genetic transformation, stress physiology and biochemistry, methylation studies, molecular markers, and many more. Sir has more than 50 research publications to his credit in reputed journals. I would, on behalf of GVS, I invite Sir to please deliver the talk. Please, sir. No, sir. No, you're on mute, sir. So you're not audible to us. So you're not audible to us.
Direct laptop. Hello. Hello.
Yes.
Yeah, now I think audible, but
Yes.
you will get slow voice because I am directly using laptop speaker.
No problem, sir.
Okay. Okay. Thank you, madam, for introduction, and I thank Gujarat Vigyan Sammelan team for providing the opportunity to present the work. And just I share the slide. Just I think voice is okay, right?
Yes, sir.
Yeah. Can you see the slides?
Yes, sir. Yes.
Thank you. So the talk of my age, genetic engineering for abiotic stress tolerance using plant genetic resources of the slime habitat. See why I selected this topic for today's talk, because we are situated in Gujarat, and Gujarat have a long slime coastline. So we are using the plant resources from that area, I mean line area, for genetic improvement of different crops. So today we will be discussing about the work of transgenic developments in CSMCRA. So basically halophytes. Okay, what are the halophytes and why slime plants are important for breeding purpose? So basically, if we talk about the global population, then by 2050 we are going to cross 9.7 billion population, and for that we need improved food production to sustain the our food supply. Okay, just okay. And if we go back and we check, then in 1960s through Green Revolution we got a good increase in our production of major green crops, and through that revolution we got many elite varieties through breeding, hybrid development, and through fertilizer application and advancement of crop management. But now if we talk in present scenario, then there are number of stresses like biotic stress, means insect or pest attacks, and various abiotic stresses like temperature, water, salinity. There are many more stresses which affect the crop production. And if we talk about the salt affected area, then globally we have 1030 million hectares of land which is salt affected as per FAO data in 2015. And we know our major crops are glycophytes, means they are not able to tolerate the salt. So basically they falls under glycophytes. So major problem of today's scenario is increasing salinization of agriculture lands, because we have glycophytes as a major crops, and day by day we know that salinization of agriculture land is increasing. But we have an advantage of slime plants, because the plants which grow in salt, they have some transporters or they have number of genes which maintain the iron homeostasis in halophytes. Means slime land or slime environment, they cause iron toxicity or nutrient imbalance for plants. But halophytes, they have some mechanisms, they have some genes or specific genes which make them able to grow under slime condition. And there are number of transformation studies and number of successful studies, and well-known successful stories are herbicide and insect resistance. Okay, so today we will be discussing halophytes or seaweeds as a genetic resources, and today I will target only two genes. We will focus only two genes. One is antiporter gene from Kappa ficus alvarezii, means sodium hydrogen antiporter, which is taken from Kappa ficus alvarezii, which is a red seaweed, and another is Seliconia brachyta gene, which is RNA polymerase third complex 5-like subunit, means SBRPC5L. So these are the two genes we will be discussing today. Now, before starting talk, just we should know the basics. And nowadays everyone have in 4G in our mobile. But see here, for crop genetic improvement we have 5G. Okay, so what are 5G? Means resources from where we can go for crop improvement. If we have genome assemblies, for given crop we can go. We have germplasm characterization approach. We have genes which can be identified and used for genetic improvement. Then we have genomic breeding technologies, okay, using our existing knowledge. And now recently we know genome editing or gene editing technologies for improved plant performance. So we have different G's here for crop improvements. And our halophytes, means plants of sline length, they occupy very specific position in these 5 genes. They are very unique source of genes which provide abiotic stress tolerance in general and salinity stress tolerance in specific. So this is just about a specific. Then there are ways how we can improve the crop productivity. Okay, so what are the different ways we can improve the disease tolerance? Okay, by improving disease tolerance, we can improve the crop growth or crop yield. We can improve the flood survival. Okay, plant ke andar flood survival we can improve. We can get, then we can improve the water capture capacity. We can target improved salinity tolerance. That is today's focus, where we will be using genes to improve the salinity tolerance. Then we can improve the seed filling properties in crops. Okay, we can optimize the photosynthesis, or we can improve the photosynthesis. We also worked on photosynthesis. We cloned some cytochrome genes and used them to improve the photosynthesis in crops. Then we can also improve the desiccation tolerance or water stress tolerance, or desiccation avoidance. This is also related with salinity, because some salinity also cause physiological drought. So salinity and desiccation tolerance, they are almost correlated with each other. Then we can improve the nutrient uptake in roots. So these are the different ways, or these are the different targets. Manipulating these, we can improve the crop productivity. And today we will be using genes from seaweed and halophyte. One gene from seaweed and one gene from halophyte. And then we will be discussing the transgenic of those crops, those plants. So let's see. Gene, when we are talking about seeds, then we are working on Kappa ficus. So Kappa ficus is a red seaweed, and we done some transcriptomic studies on this red seaweed, where we identified approximately 18% novel gene, unique genes. Unique means they are not showing similarity with the public database. जो public में है, उनसे match नहीं कर रहा है, means they are totally new, okay, uncharacterized thing. So from their pool, from this pool, we are using genes, and we are characterizing those genes for their role. So from these data, we are not discussing today transcriptomics. So from these data, we used one gene, that is NAH antiporter, this blue color gene in yellow box. So we will be using today or discussing sodium antiporter gene. Then this is how we cloned the gene. We have taken the Kappa ficus, then we extracted the total RNA. From RNA, we synthesized the cDNA, and from that cDNA, we used some degenerate primers, and we cloned the gene partially. Okay, and from that partially cloned gene sequence, we performed 3' rapid amplification of cDNA ends and 5' rapid amplification of cDNA ends. So through 5 and 3' rays, we amplified the gene, and we got the full copy of gene. Okay, and then that gene was cloned in plant transformation vector. Okay, this is P-Cambia 1301 vector. So we cloned that gene in P-Cambia. So these are the basic steps of gene cloning. Okay, I will also repeat these steps in second gene, but with slightly fast speed, because we have time limits. Okay, so this is just an in silico analysis. We will not go in detail, but basic information of gene is the clone gene is 1071 base pair long, and it has coding region of 746 base pair. Okay, and this gene encoded a peptide of or protein of 251 amino acid. Basically, when we divide 756 with 3 number of okay, then we get 252. So one is stop codon, and otherwise protein is 251 amino acid. So this is about the basic information of sodium antiporter gene, which encode the antiporter protein, and that is membrane localized. Okay, this is in silico analysis, means on computer system. Then we have second gene, and that second gene was taken from Salicornia. Many of us know we have a very good plant, especially Salicornia, that grew totally in saline water. Okay, where seawater regularly comes and go, means intertidal regions. There it grew, and it grew luxuriantly with very good growth. Okay, you can see the photographs. So this is a plant which have ability to tolerate very high salt concentration, and from that we also developed EST database, and among that database 30% EST were novel, means not known. Okay, so from this database we have taken one gene, and that gene was this. Okay, this was earlier, it was not known, so only a accession number is given, EB484 674.1. So it is a accession number because it was earlier not known. So we have taken this gene and we cloned. It was cloned same, RNA was extracted, cDNA was synthesized, then from partially we performed the race, and then finally we get the full copy of gene. Okay, so this is the gene. Then we sequenced this gene and we blast this gene with public database, and that database, from that database, the database showed that it is a RNA polymerase third complex 5 like subunit gene. So we named it same RPC5 like subunit gene. So this is novel genes, means not matching with other existing database. So this is again. Then we have taken both genes and we checked, okay, where the product of these genes is localized, means subcellular localization. For that purpose, localization, we have some vectors and specific known as p-enter d topo vectors. Okay, so in this vector we have some sequences which produce fluorescence like thing. So we cloned our gene in this vector, and then we transformed the onion epidermal cell, and we observed the fluorescence under epifluorescence microscope. So where we get the fluorescence, then we confirm that this is membrane localized, and another gene is also membrane localized, means the protein which is encoded by these genes is working at membrane location. Okay, so this is subcellular localization of both genes. Then we constructed the transformation vector. We used the pCambia vector 1301, and then we mobilized those vectors or those construct in agrobacterium, and then we co-cultivated the agrobacterium with tobacco disc. So co-cultivated, and then we regenerated the transformants. So during this regeneration, we also use some antibiotics. That antibiotic allow only regeneration of positive transformants. Okay, so we regenerated, we elongated, then we rooted, then we transferred the rooted shoot in small pots containing sterile soil. Then the plant was transferred in bigger pots, and then finally under containment facility where they flowered and produced the seed. And until last, we harvested the seeds from transgenic plants. So this is how we generated the transgenic plants. Then okay, and before going to analyze their performance under salinity, we have to check whether the seed which we have, they are transgenic or not. For that, we perform different assays. PCR assays means we grow them and we extract the DNA, and then we perform the PCR analysis where we amplify the GUS genes, where we amplify the antibiotic gene, or we amplify our own specific genes. Okay, just antiporter gene or RPCL5 gene. Okay, so we check the transgenics key, whether they are positive or negative, and then we select only the positive one, and we go for further analysis. Now we have the positive seeds. So we have to check at germination stage. Okay, so germination, how we perform the germination studies, we just take the seeds from transgenic plants and then we tested their germination on control condition and then salt condition and under osmotic stress. There we record the germination percentage and then we record the seedling growth. Like in second portion you can see the seedlings. So we record the seedling growth, where we record the fresh weight, we record the dry weight, and then we also calculate, based on dry weight, we also calculate the tolerance index. Okay, so in these tests, transgenic lines, they perform better. Seed germination, it is also higher. Seed growth, it is also higher in transgenic compared with control plant. Then we see the tolerance index, it is also higher in transgenic plant compared with control or non-transgenic plants. So these are the results about germination studies. Then from that, we also record the fresh weight, it is also better. Okay. Then dry weight, it is also better. And water content means it is simple, very simple. Deduct dry weight from fresh weight and you will get water content. So when both are better, then water content will also be better. Okay, then from dry weight, we also check how that transgenic line is accumulating sodium, or it is accumulating less or more sodium. So sodium, here you can see in this graph, sodium, it is lower content of accumulation of sodium in transgenic lines under salt stress. Okay, in PEG there is no sodium, so all lines are performing equally. Then we see the potassium. Basically under salt conditions, sodium compete with potassium. So plant do not absorb potassium, but in case of transgenic, sodium compete less. So plant is able to accumulate more contents of potassium under salinity stress. So this is about how plant accumulates sodium and potassium, and then we calculate the ratio of potassium and sodium, because the ratio of potassium and sodium is an indicator of stress tolerance. Okay, from there we can get an idea that our transgenic lines are performing better or not. Then after seed germination studies, we study the behavior of transgenic plants at maturity stage. So we grow the plant for 5 weeks, 6 weeks, depending on the crop plant, and then we treat them in liquid medium, that is in hydroponics, and we give them for immediate short term salinity exposure. For that we treated them 24 hours. Okay, we gave treatment of 24 hours, so immediate plant will respond, and that responses we will capture in our studies. So how relative water content changes. See, relative water content of transgenic plants is better as compared to control or vector control plants here in under both salinity or PEG. Then we check the membrane leakage, means when we treat with salinity, then membranes lose some property and it lose some solutes. So that we check with, it is very simple test. We use EC meter and then we perform some analysis. So here you can say, see, there is significantly low electrolyte leakage in transgenic plants in second graph. Then why this leakage is there? Because membrane lose some properties, means membrane, they are made up of phospholipids. So we check the phospholipid capacity, means performance, means membrane lipid peroxidation. Okay, so we check the lipid peroxidation, how lipid peroxidation is in transgenic plant, and that is tested spectrophotometrically by measuring or determining the MDA content. So when MDA content is less, then membrane damage is less. So here you can see under salinity treatment, the transgenic lines are showing less lipid peroxidation, means they are performing better at cell membrane stage. And then alternately, we also perform the membrane stability index. When lipid peroxidation is less, then electrolyte leakage is less, then there should be membrane index should be high. So we also calculate membrane index. See here you can see in transgenic plants, the membrane stability index is significantly higher under stress condition. Okay. Then we also check the solute potential, how the transgenic plant is able to accumulate the solute. Okay, so we check. Then solute potential, it is always lower in transgenic plant as compared to control plant. The graph, because we know the solute potential is always negative. So when it is more negative means okay. Then we also check the same parameters we check in second. So I will not be discussing this because the same parameter in second transgenic line. Okay, there's a relative water content, then electrolyte leakage, then we correlated with lipid peroxidation, then we correlated electrolyte leakage with membrane stability, then solute potential and hydrogen peroxide. Hydrogen peroxide I will again speak in next slide. So here I am skipping. Okay, so then why the toxic effect of salinity stress is there? Because the stress, they generate the reactive oxygen species in plants and they harm the different processes. So we detected the formation of superoxide radicals or the formation of peroxide radicals in plants. They are very simple assay, lab assay, okay, and NBT assay. They are very simple assay performed under laboratory condition and they are qualitative assay. So here you can see there is less accumulation of peroxide radical in transgenic plants under salinity stress. Here you can see less accumulation of that superoxide under salinity or PEG stress in both lines. One is antiporter gene transgenic line and second is RPCA5L transgenic tuberculin. So in both lines, the accumulation of reactive oxygen species is less compared with control. Why this this accumulation of reactive oxygen species is less? Well, there should be some mechanism. Okay, for that we check the expression of antioxidant genes. Okay, there are number of antioxidant enzymes. We know superoxide dismutase, catalase, ascorbate peroxidase. Okay, peroxidase enzymes, they sequester the reactive oxygen species. So we can perform enzymatic assay as well as we can check the expression of those genes. Okay, so we check the expression of sodium superoxide dismutase gene. So the transgenic lines, they have better expression of those genes, SOD gene. Then we check the catalase expression. So catalase expression and ascorbate peroxidase or peroxidase expression is always higher in transgenic line under stress condition as compared to control. So when enzyme is higher, then it will sequester more superoxide radicals, more peroxide radicals. So there will be less content of reactive oxygen species in transgenic plants under stress condition. And when there will be less reactive oxygen species in transgenic plants, then they will have better membrane properties or better membrane functioning and better tolerance. So this is about the antioxidant expression. Then after antioxidant expression, ROS expression, we have to check the photosynthesis or photosynthetic properties of the transgenic lines. Okay, if they have better photosynthesis, then definitely they will have better productivity or better yield. For that we perform the senescence assay. Okay, for that it is just similar assay like we have in PGR, cytokine enrichment and lag effect. So similar kind of assay is there. So we take the disc of leaf and then we grow those leaves in different stress treatments. Okay, so how they visually appear means how faster they lose the chlorophyll pigments, how faster they develop the visual observations or visual symptoms. Based on those, we get the idea about the tolerance of those transgenic lines for particular stress. And then here you can see in transgenic lines, okay, this is a wild plant, means control plant. Here you can see first, second, third, fourth, and fifth. Fifth is totally yellow. But in case of transgenic line C L6, still the fifth veil is maintaining its vigor or its greenness. Similarly, in L15, you can see they are still maintaining their chlorophyll pigment. So they are able to retain chlorophyll or photosynthetic pigment for more times under stress condition. When they are able to retain more pigments for more duration, then definitely they will perform. So we also measure the total chlorophyll and carotenoid content they are also better. Means this senescence assay, it is qualitative, and then we perform the quantitative assay also. So chlorophyll and photosynthetic pigment, they are better. Then this is same assay for second gene from halophyte, that is silicornia. So we are not going to discuss, but yes, we can also estimate the chlorophyll A, chlorophyll B, total chlorophyll, carotenoid, and we can also interpret results with visual observation, and we can also calculate the ratios of chlorophyll A, B, and total chlorophyll and carotenoid. They have different rules. So from basic data, we can calculate those parameters also. Then we perform when pigments are better, then transgenic lines should perform better photosynthesis. So we perform the IRGA estimations, infrared gas analyzer. We have machine. With that, we can measure the photosynthetic gas exchange. So here you can say first graph, it is net photosynthesis, means how much carbon is fixed by this plant. Or so here you can see under stress condition, salinity or PEG stress, the rate of net photosynthesis is higher under stress and PEG condition. Then we can also measure the stomatal conductance, means gas exchange. Okay, it is also better, because when photosynthesis is better, then there should be background processes should also be better. So stomatal conductance is also better. Then in leaf, the concentration of CO2, means intracellular CO2 is also better. Then transformation is also better in transgenic lines, because stomatal conductance is better, then stomata remain more time open, so definitely it will lose water. So transgenic transpiration is also better. With net photosynthesis and transpiration, we can also calculate water use efficiency of plant. Means if we divide net photosynthesis with transpiration, then definitely per molecule of water, how much carbon is fixed, we can get the idea. That is water use efficiency. So this is just an idea for your information. Then we perform chlorophyll analysis, means fluorescence. Okay, chlorophyll fluorescence analysis. So from that fluorescence, we can calculate the photochemistry, PS photosystem to photochemistry. So this photochemistry is better. We can calculate electron transport rate. Okay, we can calculate photochemical quenching and non-photochemical quenching. These are the specific parameter. Quenching means how much light is used in photochemical reactions and how much light is not used or in non-photochemical processes. So NPQ and PQ there are many more parameters, but these are the major important parameters. So all the parameters are better in transgenic lines, means our transgenic lines are performing better under stress condition, means they are performing better photosynthesis. Okay, then when photosynthesis is better, then the products of photosynthesis should also be better. So we estimated the amino acid concentration, means biochemical concentrations, biomolecules. Okay, so amino acid, they are better when contents are better. Okay, then we check the proline. Proline is stress indicator. Okay, so it is also better in transgenic lines under stress condition. Then polyphenols, they are also very important molecules. They sequester the reactive oxygen species. Basically, they are non-enzymatic antioxidants. So the contents of proline is also better in transgenic lines. So they help to sequester the reactive oxygen species efficiently. Then these are the sugar and starch. They are the direct product of photosynthesis. So soluble sugar content and reduced sugar content, they are better. Their amount is better in transgenic lines. Then when sugar is better, then there should be better content of starch synthesis. So we also get the better contents of starch. Then this is same free amino acid, proline, starch, and reducing sugar. Okay, this is in same in second gene. So I am skipping this slide. Then this is a simple assay for starch localization, where we similarly, some of you are studying in BSc. So we have, starch test. We will take potato and then stain with potassium iodide. Similar kind of test is this. Very simple test where we can detect the accumulation of starch in our leaves of transgenic lines and control plant. Here you can see clear difference in control plant. This is control plant, this is control plant. So there is less staining, and here you get some staining of that potassium iodide. So we can say transgenic plants accumulate better content of starch as compared to this. So this is a qualitative assay for starch estimation. Then we perform, these were the assays of transgenic plants given short-term exposure of stress. Then we perform long-term stresses, okay, where we grow the plant and we treat plants for weeks, okay. So you can see here we treated plant for 21 days, means 3 weeks. We provided or we irrigated the plants with NaCl 200 millimolar NaCl for 3 weeks, and we irrigated plants with PEG solutions for 3 weeks, means alternating day or after every 2 days, like that kind of treatment we provide. And then we tested the recovery of those plants. So in transgenic plant, you can see better recovery of plants under PEG stress, okay, means osmotic stress. They perform the better, so the better recovery after stress period is over, okay. Then in long term, we tested the plants pigments, because pigment are the major player in plant system for photosynthesis. So we estimated the chlorophyll pigments and we estimated the carotenoids pigments in plants treated for 4 weeks, 8 weeks, and 12 weeks of stress, okay. And we found that the transgenic plants have better contents of these pigments. Then when pigments are better, then it gives you an idea that when pigment is better, then photosynthesis must be better, and products of photosynthesis must also be better. So we tested we estimated the soluble sugar. They are also better in transgenic plants as compared to control plant after 0, 4, 8, and 12 weeks of stress. Then we also checked the photosynthesis of those plants which were exposed for longer duration of stress. So we observed photosynthesis after 12 weeks of stress, okay. 12 weeks we provided them salt and osmotic stress, and then we determined the photosynthesis. So photosynthesis, we observed it is better. Gas exchange, it was variable, because under field condition there are so many factors. So stomatal conductance, it varied, and then transpiration, it was always just higher in transgenic lines as compared to control plant under stress condition. Then we checked the photochemistry. So photochemistry, it was in few lines, it was better in transgenic lines under stress condition, and under osmotic it was variable, it was not significantly different. But at the same time, when we assess the photosynthesis, electron transport rate, it was higher in transgenic lines as compared to control lines, okay. So this is about, then we check also we measure the photochemical quenching and non-photochemical quenching. We estimated different parameters, so transgenic lines perform better. Now it is the last slide and take home message that definitely in future our 5G approaches or 5G breeding approaches is going to change our future of plant science or breeding, and in that our halophytes or seaweeds, means the plants which are growing purely with seawater, they are going to play major role, okay. And the genes which we discussed today, sodium hydrogen antiporter gene or ribosome polymerase third complex 5 like gene, they are both genes are potential candidate gene to be used in breeding programs for development of climate resilience crops, okay. So this is the final message of today's presentation, today's talk. And then in last, I thank CSIR, our parent organization. Then I also thank Gujarat GSB team, Government of Gujarat, for financial supports, and then CSIR or UGC for supporting students in different forms, and SESIR for allowing them to register in PhD. Then thanks to parents, seniors, and junior friends. And I also thank Gujarat Vigyan Sammelan 2022 team for providing me this platform. And in last, I thank the persons who are behind the conceptualization of concept of Azadi Ka Amrit Mahotsav, that give opportunity to all of us to interact with each other. Okay, thank you, thank you.
Thank you so much, sir. we can quickly take 1 or 2 questions from participants. Any questions? Since we have no more questions, on behalf of GVS, I would like to thank you, sir. It was wonderful talk. Thank you, sir.
Thank you, madam. Thank you.
Our next talk is related to plant biotechnology again. I would like to invite Dr. Rukam Singh Tomar to talk on genome editing for crop improvement. Dr. Tomar has done his PhD from Gujarat Agricultural University and is presently working as Associate Professor at Junagadh Agricultural University, Junagadh. He has research experience in fields of marker-assisted selection, plant breeding, transcriptome sequencing, genome sequencing, and QTL mapping. He has more than 100 research publications to his credit. On behalf of GVS, I invite you, sir, to please deliver the talk. Please, sir.
Good afternoon to all. Whether I'm audible?
Yes, sir.
Okay.
Yes.
Okay. Is my screen visible?
Yes, sir.
Okay. Thank you for giving me this opportunity to talk about genome editing and to show the work which we are carrying out at Junagadh Agriculture University. So before starting my presentation, I would like to introduce my university. I come from Junagadh Agriculture University and I'm working at Department of Biotechnology. In Department of Biotechnology, we are working on genome and transcriptome sequencing. We have sequenced many genomes like cumin, coriander, fennel, dill, scalcium rosea. So we are first to report the genome of all these kind of plants and few microbes which are important to agriculture. We have also carried out transcriptome sequencing in different crops like bajra, castor, chickpea, and coriander for different traits. For example, in bajra, we have carried out transcriptome sequencing to identify the transcript responsible for drought tolerance, similarly in chickpea for salt. So this is different kind of work which we are doing at Junagadh Agriculture University. And recently, we have started working on genome editing. The basic work at Junagadh Agriculture University is to improve the crops. So if you want to improve the crop, so normally we go for breeding. So if you want to improve the crop, there are different methods for improving crops like domestication, selection, and hybridization. Here you can see the tomato plants. Initially, tomato plant where the size of tomato was very small, and we have improved those tomato plant to the bigger size of tomato. Here you can see the maize. This is maize plant. It has been improved to initially the maize size was very small, and we have improved the maize plant by using different methods like domestication, selection, and hybridization. Normally, we use hybridization when the variability in the plant is not available. So we try to create variability in the plants by using hybridization. Even after hybridization, the variability is not available in the genotype. In that condition, we use mutation. So mutation is a technique to change the phenotype of a particular individual. So it is a sudden heritable change in the phenotype of an individual, and this change could be in the gene, in the chromosome, or in the plasma gene. But normally with this mutation is being created by either altering the DNA of nuclear DNA or by altering the cytoplasmic DNA. And normally we carried out mutation by using physical mutagens and chemical mutagens. So by using these mutations, we have created a lot of variability. You can see the tomato, we have created a lot of variability in tomato. We have created citrus, which are seedless. and we have introduced new colors in chrysanthemum by using mutation. So this is the beauty of mutation, to create variability where it is not present. But there are some drawbacks of this mutation. The basic drawback is these mutations are rare and they are predominantly recessive. So these cannot be used in breeding. And the basic limitation of mutation is they are random and unpredictable. So if the mutation is random and unpredictable, then it is of no use. But now, if you are able to do mutation, and that is precise mutation, then it can do wonders. So now the new technology is coming up that is known as genome editing. So in genome editing, we can do precise mutation, a mutation at a particular place. So before moving to that, what is genome editing? So it is an ability to change specific genetic in the genome. It could be at specific location, or it could be of specific alteration, or it could be specific to particular gene. And this was normal in bacteria and yeast and mammals. And here we take advantage of the process by which DNA is being repaired. So we take advantage of that and we use genome editing for specific mutation. So what is basically required if we want to do specific mutation? The first thing is you need to have a specific DNA sequence at which you want to carry out your mutation. And second thing is the nuclease which can cut that DNA. So basically you need only two things. One is the sequence of the gene which you want to mutate, and a nuclease which can cut that gene. So normally this is being carried out by two methods. Either it is by recognizing the sequence which you want to mutate by proteins, or recognizing the sequence which you want to edit by RNAs. So today I shall be talking about on RNA recognition of DNA. So basically this method is not new. It is being used in bacteria, and this method is known as CRISPR-Cas, which stands for clustered regularly interspaced short palindromic repeat. So here in bacteria there are different repeats which are present in the bacteria, and in those repeats there is a spacer, and these spacers are specific to bacteriophage. So in bacteria there are different repeats, and between these repeats there is a spacer, and these spacers are specific to bacteriophage. So here you can see we are having a bacteria, and that bacteria has been infected by bacteriophage, and this bacteriophage will release phage genes in the bacteria, and these phage genes are being recognized by the they have a copy of that in the spacer region. And once the bacteria is being infected by bacteriophage and the phage gene is being released, these CRISPR region are transcripts and they produce CRISPR RNA. And this CRISPR RNA have the ability, because they have a guide RNA, they have the ability to recognize the phage gene, and they recognize, and it is being cut by these CRISPR RNAs. And this is how the bacteria gets the immunity against the bacteriophage. Here the basic role is of Cas9. What is Cas9? It is a protein which has an ability to cut the nucleic acid, and it cuts at double standard break, and this Cas9 is being directed by CRISPR RNA. It is being directed by the CRISPR RNA, and this CRISPR RNA can be specific to any sequence. So by using this method, you can edit any gene which is having the sequence, and if we are able to generate the guide RNA specific to that sequence, and using Cas9, you can edit any gene. So normally now the bacteria which is using this CRISPR technology for developing immune in themselves has been being used as an editing tool, and this editing tool is basically based on the fundamental that it breaks the DNA at a particular site based on the guide RNA, and this cut in the double-stranded break in the DNA is being repaired by the DNA, and while the process of repairing, there are some errors. And based on that error, the function of DNA is, the function of that gene is disrupted, or the gene is being knocked out. Normally, the DNA try to repair themselves in two methods. Either it is non-homologous end joining, and non-homologous end joining, they try to join the non-homologous ends, and while doing so, there is some addition or deletion in the sequence, and this disrupts the function of that particular gene. And if we want to, the other method is that homology directed repair. In this condition, we need a sequence of a donor gene which is responsible to direct the repair. In this condition, you can insert the gene into that breakage. So there are two kinds of method by which you can disrupt the function of that particular gene. So if we want to use this technology to edit the plant, there are three things basically required. One is Cas9 protein, other one is guide RNA, and third one is donor template DNA. That too, if you want to insert a particular gene, or if you just want to edit the gene, in that condition, you just need Cas9 and guide RNA. And there are two methods to do so. One is transgenic method, and other one is non-transgenic method. And if you are using transgenic method, in that condition, you just need to physically insert Cas9 and guide RNA into the genome. And that cell which is transgenic, or in which you have transferred the Cas9 and guide RNA, need to be regenerated using tissue culture technology. And later on, once the plant is being developed, the transgene can be eliminated once it has been edited. And in non-transgenic method, same Cas9 and guide RNA can be inserted into the cell, and the gene and the cell can be regenerated into the complete plant by using tissue culture technology. And the protein Cas9 and RNA which we have inserted into the cell are naturally degraded over the time. So there are two methods, transgenic method and non-transgenic method. If you are using transgenic method to do so, in that condition, and if you just want to edit the particular gene, in that condition, you need a construct which has only Cas9 and guide RNA. And if you want to insert some gene into it, then in that condition, you need to have a construct with Cas9, guide RNA, and a donor template which you want to insert into it. If you're using transgenic method, then in that condition, once the edited plant is being developed, you need to remove the transgene which is present in the transgenic plant. In that condition, if it is a field crop, you can use crossing, inbreeding of the lines, and in the segregating population, or by using back crossing, you can remove the construct which has been inserted. And there are some other molecularly techniques by which you can remove the construct which you have inserted for editing. And if you're using non-transgenic method, then there is no need to do anything other than need to do anything. And there are different techniques like particle bombardment, protoplast, and viral replicons by which you can develop non-transgenic edited plant. So this is how normally plants are being improved by using genome editing technology, that specifically CRISPR-Cas. Now I will show a small example in our lab by which we have developed a groundnut edited plant. So what we have done in our lab is we have tried to modify groundnut plant for FAD2 gene. Basically, FAD2 gene is a gene which converts oleic acid into linoleic acid. Oleic acid is good for health. It provides stability to the oil of groundnut, but this oleic acid is being converted into linoleic acid, and this linoleic acid produces rancidity in the soil. So basically, we need a groundnut variety which is having high oleic acid and high OL ratio, or oleic and linoleic ratio. So normally the variety which are prevailing in the market, for example, GG20, GG22, or any variety which is present in the market, they are low in OL ratio. Since they are low in OL ratio, the oil quality is not that good. It produces rancidity, and it is okay. It is good for health, but it is not that good for health if it contains high oleic acid. So there is a gene known as FAD2 gene, and this gene converts oleic acid into linoleic acid. If somehow we are able to mute the function of this gene, in that condition, oleic acid will not be converted into linoleic acid, and we can have a variety with high oleic acid. So this was the basic concept behind doing this. So what we did is, so there are different steps to do so. The first step is to have a guide RNA designed from the targeted sequence. Here the targeted sequence was FAD2 gene, and we need to try to avoid off-target effects and assembly of Cas9 and guide RNA, and that will be and we need to have an agrobacterium transformation to deliver this construct into the plant, and tissue culture. From tissue culture you need to develop the complete edited plant, and later on you need to confirm the plant whether it has been edited or not. So here you can see this is the complete sequence of FAD2 gene. This is the complete sequence of FAD2 gene. And there are a lot of software for developing the CRISPR, developing the guide RNA. So this is, for example, CRISPR-P is there, ChOP is there. Here we have used CRISPR Direct software to develop the guide RNA. So what you need to do is you need to put your sequence of FAD2 gene into this, and you need to click a simple button design, and you will have a complete list of guide RNAs from that particular gene. Here you can see number of off targets are not presented over here, because the time we carried out this research work, at that time the genome of peanut or groundnut was not available, so we cannot detect the off targets over here. But for that I have taken an example of rice. Here you can see in rice this same software has been used, and here you can see these are the guide RNA, and here you can see the off targets which are there. So normally we need to get a guide RNA which has less number of target. For example, here you can see there are 3 guide RNAs which are having 1 and 1 off targets at 20 mers, and 12 mers there are 70, 14. So we need to take this guide RNA for further work, because it is having less number of off targets. So this is the FAD2 gene, and from this FAD2 gene we have taken this as a guide RNA, and this guide RNA was used in the construct and to edit that particular gene. So this is the guide RNA, and we have procured the vector from AD gene. This is the vector from the AD gene, and this vector has Cas9. The only thing we need to do is we need to insert our guide RNA into this vector. So what we have done, we have taken this vector. This is our guide RNA ligation, and through ligation, our guide RNA was inserted into this, and that was cloned into the E. coli, and from E. coli the plasmid was extracted. It was checked for its quality, and later on it was again transformed into the agrobacterium, so transferred into agrobacterium. So before going for cultivation of this groundnut and agrobacterium, we need to check whether the agrobacterium has the plasmid or not. So for that, we have designed different primers specific to the Cas9 and specific to UC6 promoter and 35S promoter, and we found that okay, the agrobacterium has the construct which we need to transfer into the plant. Now the agrobacterium and the cotyledons of groundnut were co-cultivated in this, and from those cotyledons the complete plant was generated. So here you can see we have used tissue culture protocol to generate the complete plant from cotyledons, and this shoot was generated from this. Near about we have generated near about 150 shoots from that co-cultivation of agrobacterium and groundnut cotyledon, and from this the DNA was extracted. DNA was extracted to find out whether the Cas9 integration is there or not. So here you can see again we have extracted the DNA from the groundnut shoots, and we used Cas9 specific primers to find out whether the integration of Cas9 has taken place in the groundnut or not. And we found that in, for example, plant number 1, 2, and 3, Cas9 integration was there, while in 4 and 5, Cas9 integration was not there. So this is how we found that Cas9 integration was there. So the positive plants which were having the Cas9 integration were further moved for rooting and hardening. So here you can see the complete edited plants are being grown in the greenhouse. So these are all edited plants, and these plants were harvested and the seeds were checked for, and the plants were harvested before plants were harvested for seeds, and each and every plant were again DNA was isolated from those plants. All the edited plant, we were having 27 edited positive edited plant, and the primer were designed from the FAD2 gene to find out whether the editing has taken place or not. So all the 27 plants were amplified for this gene, and they were sequenced using Sanger sequencer. And here you can see this is control plant and this is groundnut edited plant. So here you can see there is an addition of A. There is an addition of A in the edited plant in comparison to the control plant. Over here you can see this is the palm sequence. After palm sequence you can see it is TCCAC, and here you can see TCCAA. So A has been added in the edited plant, and because of that the complete frame shift had taken place. The complete frame shift has taken place. So all the 27 plants were edited. That was confirmed they are edited. But whether the FAD2 gene has been knocked out or not, for that we went for OL ratio analysis of all the harvested plant, and we found that the ratio of control groundnut was 1.97, while the ratio of edited plant was 5.39, 5.79. All the edited plant were having higher ratio than the control one. And we also matched the morphological difference between the seeds of GG20 control plant and the edited GG20. So there was no change in the morphology of the seed, but the OL ratio has changed. It was 1.97 and it has reached to 5.79. So this complete research work took 1.5 years to do all these kind of things. So in a period of 2 years, we were able to develop GG20 with high oleic ratio. If we have done same thing using traditional breeding method, it would have taken 10 or 12 years to develop a GG20 with high oleic ratio. So this is how you can use new technological tools like CRISPR-Cas or some other TALEN or some other editing tools to improve your genotypes. So this is so basically, if you want to improve anything, in a short duration, you can use editing tools to do so. We have also carried out a research work on tomato. In tomato, we have targeted a gene that is known as ALS gene, and primarily this gene is responsible for action for at least 5 classes of herbicides. So the basic idea was, if we are able to edit this gene, the plant will become resistant to herbicide and So for this, what we did is, this is the same flow. What we did is we took the ALS gene from the database of tomato, and again we designed the guide RNA for tomato. And again, we have procured this construct from Edgene, and this construct was having the Cas9, and we just inserted the guide RNA into it, and a complete tomato protocol was developed, and we were able to have an edited plant. And this time we have not sequenced the complete gene, but we sequenced only the guide RNA. So here you can see, this is the control. There is a change in the CA. Here there is a change in the CC. So again, the gene for ALS has been edited. So this is how you can use. But this work is still in the progress. We are in the process to spray the glyphosate and to see whether the plant are able to tolerate glyphosate or not. So that process is in process. So this is how you can use genome editing to improve the crops. Different scientists in different places of the world have used this technology to improve the plants. For example, in wheat, powdery mildew resistance has been developed by editing the EDR1 gene by using CRISPR-Cas method. In tomato, SIMLO1 gene has been edited to make it resistant to powdery mildew. Tomato again has been edited to make it parthenocarpic, and similarly ALS gene in rice has been edited to make it herbicide tolerant resistance. And in maize also, a gene known as ARG-S8 has been edited to make it drought tolerant. So these are the few examples where the crops have been edited for different traits. And similarly, if you do same thing with traditional method, it takes years. So this is how you can use genome editing for crop improvement. So I think this is all from my side. And so the basic importance of this technology is that it is site-directed modification of new product. It reduces time to get product, reduce the number of plant involved, and a longer DNA sequence can be inserted into this. So this is all about from my side. If you have any questions, let me know.
We can quickly take 1 or 2 questions from the audience. Thank you very much, sir, for the talk. Any questions from the participants? If you have no more questions, on behalf of GVS, I would like to thank you, sir, for it was really a nice piece of work. Once again, I would like to thank all the eminent speakers for sparing their time, and also thank all the participants for listening patiently, and the members of GVS. All the members are requested to assemble back in the virtual room for next session at 2:20, and those associated with presentation in human and animal healthcare need to be on this link, and also bio ideathon, whereas those associated with industrial and environmental and plant biotechnology can join virtual room B after lunch break. Thank you. Thank you all.